So let's get started. Sorry about the delay. We actually have a busy, program designed for all of us. I want to thank you all. My name is Guigan Zhang. I'm a current president of the SFB, also a professor endowed at University of Kentucky. Today, I'm very honored to co-host this workshop with my colleague at FDA, Tina. I will hand the podium to her in a minute. So I want to just give you a couple of minutes of a background or the genesis of this whole idea. As we know that every time we talk about FDA, talk about regulation, there's always issues that, different sides have a different story. From industry, they just feel like the whole process is holding back the innovation. And then from the FDA side, as you know, that their question is, do we have enough technology or tools or even methodology or information to protect the safety, the public safety? So this is basically the genesis, the starting talking about, and then Tina and I, and also a couple of my colleagues at FDA, including Frank and Henny. We've been working towards this for almost the full year and maybe even longer than that. So the idea is basically just say, how can we change the mindset and then to inject a new perspective or even new way of working together, how academia and regulator and business industry, even consultancies and things like that come together and work as a team and find the best way to go forward. So a couple of questions we hope that we'll start to ask, and then we will have a follow-up. This is just the beginning of the whole process, is that what can we do to enhance our collaboration, and also how to develop methodologies, new methodology based on new way like in vitro, even in silico, and also how to convey those innovation into tools that FDA can actually use and also industry will embrace, all of those. And then the last issue is how can we also bring this to the education side? And how can we prepare more future leaders to lead us in this area? So this is just my kind of a 2 minutes capture of the whole genesis, and now I'm going to turn to my co-host Tina. Tina, you want to take over?
Yeah, good morning. Can you hear me okay there?
Well, I don't know if a room can hear you. I mean, I'm only using my, maybe I can use this. Try say it again.
Good morning. Can you hear me?
Oh, yes, very good.
Oh, great. Awesome. I can also hear the audience there. It's nice to connect with you virtually during this difficult time that we're still facing with the challenges of work at home. The FDA is still being very cautious in having attendees visit meetings, so I'm sorry that I cannot be there in person to meet all of you, but I am thrilled and excited to co-host this session with Dr. Zhang. Bear with me 1 second. So Dr. Zhang gave you the welcoming remarks about the Advancing Translational Science and Regulatory Science Synergistically. This is a meeting, as he mentioned, we've been planning over the last year. We're really excited to bring together speakers from all across the ecosystem. And I'm going to take a few minutes to walk you through a few key terms that we're going to use today to kickstart the conversation. And then I will introduce the agenda, our speakers, and a few housekeeping things for this morning. So it's really important that when we're talking about science, applied science, translational science, and even regulatory science, that we have a clear understanding of what we mean. So I'm just going to take a moment to walk you through these to make sure we're all on the same page about what we mean by science. You are all working in the space of science for the pursuit and application of knowledge and understanding the natural and social world, utilizing some systematic methodology based on evidence. And you want to use that evidence in a capacity that's either in an applied sense, where your applied research can result in some kind of technology or innovative discovery. This can lead to different types of products. This could lead to procedures or technologies that can be used even in the regulatory space, not as medical products per se, but even to support, say, the regulatory process. And we'll talk about that in a moment. There's also the aspect of translational science. And I know that in the space of biomaterials, translational science is really critical for helping us to translate those applied researches into what folks term as bench to bedside. In the aspect of translational science being the process of turning observations that take place in the laboratory, the clinic, or the community to really improve the health of individuals and the public. And that, of course, brings me to regulatory science, which, just to be clear, is not FDA's way of regulating science. It's actually the science of developing tools and standards and approaches that can really help support the evaluation of regulated products. And what I hope you'll walk away from today is that we're going to showcase different kinds of regulatory science output from different stakeholders in the ecosystem. And I also hope that you'll take away that regulatory science does not just benefit or come from FDA. It also has to benefit our stakeholders, by which anything that benefits our stakeholders in the regulated process can also help and benefit FDA. So we are extremely committed to advancing regulatory science at FDA. So during today's roundtable, we have broken up the morning into 3 different sessions. We're going to start with showcasing success stories on regulatory science collaborations that involve FDA working alongside academia and industry, as well as nonprofits, public-private partnerships. So stay tuned for more on that. You'll hear how different stakeholders in the medical product ecosystem can navigate regulatory science, whether it's folks in academia, in the consulting side, industry, and also from FDA. And lastly, we'll have a panel discussion where we'll discuss mechanisms for balancing the regulatory science needs of all stakeholders to reduce regulatory inertia. So we'll kickstart the morning with our thought leaders from the medical product ecosystem to discuss their success stories and how they advance innovation through creative collaborations. That's a term that's really important to us in the Office of Regulatory Science and Innovation. You'll hear from a small startup business who capitalized on FDA's investment through our regulatory science contract mechanism to develop an innovative technology. So you'll hear that from Drs. Costa and Zhu. You'll hear from a medical product manufacturer who worked alongside other manufacturers and FDA to support the technologies needed for tool development and also for standards. So you'll hear that story from Drs. Afshari and Angeloni. And FDA, who partnered alongside nonprofits, the World Health Organization, and the Gates Foundation to advance vaccine development technology that has helped with the COVID-19 pandemic. So you'll hear that story about the advances on the polio vaccine by Dr. Chumaka. What I want you to know as you hear from each of these presentations is that no one regulatory science collaboration is the same. There's no cookie cutter for these regulatory science collaborations, by which we mean to have flexibility in how we engage with the community and how we collaborate with our stakeholders. In the next session, you'll hear about different stakeholders' approach to regulatory science. You'll hear from myself talking about regulatory science at FDA, from a professor who has a clinical translational science award from NIH, working alongside both NIH and FDA, Dr. Scott Steele, and then an industry consultant and expert working for a nonprofit whose focus is bringing tools to the medical product space to support industry and the FDA. You'll hear from Dr. Klaus Romero. And in our last session, our very most dynamic session, you'll hear conversations from different stakeholders who work in organizations that have different collaboration models with the stakeholder community. We're going to be working towards a regulatory science collaborative community. And so on that front, you'll hear from several folks from the NIH, Dr. Leonardo Angeloni, Dr. Chris Saciella. You'll also hear from Klaus again, this time representing the Critical Path Institute. And you'll hear from the CEO, Dr. Susan Winkler from the Reagan-Udall Foundation for the FDA. Each of them will give an overview of their organization, and then we will come together for a panel discussion to digest the lessons learned from today and take into considerations our next steps for moving forward with a regulatory science collaborative community. I have just 2 more slides before we kick off the next session. I do want to make a special thank you to Latoya Richardson, who is extremely instrumental in helping to coordinate the speakers, today's programs, gather their bios, and share information with all of you. She works in our Office of Regulatory Science Innovation. She serves our staff as the executive assistant and project manager, and we're super grateful for her support in keeping things running behind the scenes. Okay, just a couple housekeeping. This is a hybrid roundtable discussion today. Most of the presenters are online, but several of our colleagues are in the room. We apologize for any of the typical technical difficulties you're experiencing this morning. The meeting is being recorded and will be available following the Society for Biomaterials meeting. Each session will also function as follows. You will hear presentations by the speakers, followed by a group Q&A. So we encourage you to chime in with your good questions in the Zoom Q&A box. Please do not use the chat. We will use the chat to communicate information to both the panelists and the attendees. If you would like to ask a question to our speakers, please indicate in the Q&A box who the question, which speaker the question is for. The moderators, myself and Dr. Zhang, will be reviewing the questions and ensure that we share them with our speaker. And we will also make sure that our presenters are speaking in their allotted time. So I do acknowledge we are a few minutes over time. And with that, I would like to kickstart the morning by passing the podium over to Dr. Kostya.
All right, thank you, Dr. Morrison and Dr. Zhang. All right, I think Dr. Chu is going to start the presentation, so I'll share the screen.
Thank you, Tony. Yeah, thank you all. So, can you hear me well?
Yes, I can hear you.
Okay, thank you. Yeah, before we start, I just want to say Tony and myself, we are very grateful for being invited to speak here, and thank you for the organizing committee, and especially Tina and Reagan for hosting this session. So today, what we will share with you is a little journey that we have going through with collaboration between the FDA and academia in the area of advancing regulatory science, and also using innovative approach to manufacturing products. Specifically, what we are today looking at is nanotechnology product or nanomaterials. So Tony is a system research professor at UConn and also the founder and CEO of the Dyne Pharma. And myself, I'm the lab chief working in the FDA in the Center for CDER in Office of Testing and Research. So Tony, if you can move to the next slide. This is just disclaimer. This is a view of our individual view, not representing the agency's view. Next slide, please. So before we jump into describing what we have done, I would like to just introduce some of the innovations in 2 different industries, just to set the background in terms of what we are aimed to do. So the first example, I think, is in the auto industry, where we know at the beginning of 1900s, we've seen the use of the auto assembly line to help speed up the manufacturing and to improve the production rate of the automobiles. And at the turn of 100 years later, right now we're in the phase of a highly automated and robotic predominant manufacturing assembly line, where the production rate is much higher and also the quality is much higher. So in this, behind this innovation or this transformation are a lot of other innovations, like in the AI, use of the AI to help with manufacturing. So we've seen quite a success in the auto industry over the last 100 years to transform the industry, specifically in the manufacturing areas. Now, the second example, Tony, if you can move to the next slide, is in the silicon or computer industry, where just about 80 years ago, we had the first digital programmable computer, but now we have the power of computing at the palm of our hand, which is much more powerful, but much more smaller. So the miniaturization and utilizing the new technologies introducing into the consumer product has been really a fast-paced innovation. So this is another example of innovation in the manufacturing sector, specifically in the silicon industry. Now, turn to pharmaceutical industry. What we are going through right now, it's also a transformation. Over the last 10 to 20 years, agency, FDA in particular, has been pushing for the pharmaceutical industry to innovate in the space of manufacturing. So one example of innovation in the manufacturing space is utilization of advanced manufacturing techniques, such as continuous manufacturing, as we'll talk more a lot about today. So just quickly highlight what is the difference between a traditional batch-based manufacturing and a continuous manufacturing is that in the batch processing or batch manufacturing, unit operations are separated, and each process needs to be completed before moving on to the next. And the testing and storage and the material management and the quality testing has to be completed for each individual unit operation. As you can imagine, one, the duration is really long, and also the quality is really difficult to ensure. In contrast, continuous manufacturing, material flows through the process, and the utilization of the online sensors and controls allow much more flexible and agile and higher quality capability into produce material. Now, some other advantages I'll quickly just show maybe in the next slide, including shorter time frame to develop product and the reduced cost, reduce the footprint, and also increase flexibility in the manufacturing campaign. Overall, the continuous manufacturing is a modern technology to produce a product at a higher quality, at a higher rate, and a higher throughput, and be able to meet the demand of the market. Such as in right now, the COVID pandemic, we've seen that there is a rapid increase in demand for vaccine production, and that is something that potentially can be offered by the innovative and advanced manufacturing techniques like continuous. So with that, I will turn over the table to Tony, and then he will share the rest presentation with you about the journey. Thank you.
Thank you, Xiaoming. Yeah, so going into the problem, starting off, and this is really getting into the commercialization strategy from the very beginning, was that pharma manufacturing back when we really started would be considered, say, outdated. And one of some really great quotes here. One of today's most important tools for modernizing the pharmaceutical industry is a process known as continuous manufacturing. Statement by former FDA Commissioner Scott Gottlieb and Director Janet Woodcock in 2019. But then also for the 21st Century Cures Act, which allows the FDA to issue grants to study continuous manufacturing, a technologically advanced and automated manufacturing method. So taking a step back, as an assistant research professor and then previous graduate student at University of Connecticut, we basically started a project back in 2014 on continuous manufacturing, and that was really focused on liposomal drug products. So we're going to bring you along the journey of how we, from when we started to how we became a company today. But what it really came down to is going after multiple grants, just not having really at the beginning any idea that we were actually going to form a company. It was more, just understanding fundamentals, the science, looking for different solutions to problems that were out there. And one area was the area of nanoparticle manufacturing. So after reading multiple guidance documents and really understanding, digging into the problems, what really stuck out was quality. Quality was an issue. So if we can implement different approaches, such as a quality by design approach and a risk-based analysis approach at the initial stages, and move forward with understanding and how we can improve the technology and really get to an advanced state. So our approach was then continuous manufacturing for various types of nanoparticles. And as Xiaoming mentioned, there's major benefits involved when going from a batch approach to a continuous approach. One is the introduction of process analytical technology, where you can have better control over the entire process, much higher quality on the output. You follow quality by design principles, which ultimately leads to a much higher and greater quality product, reduction in product waste, system footprint, reduction in human error. And all of this ultimately will support end-to-end manufacturing, where you can go from API synthesis, raw material input, all the way to fill finish. And that's really the later stage of where we'd like to go with this technology. What we started off with at the beginning was really just a risk-based approach. And this was to really understand the problem, but also understand the materials that we'd be using, the process, potential process steps that we'd be using. So on the left-hand side, this is an example of Ishikawa diagram. It's a really great tool. You really put down all the information you can gather from literature, from conferences, and speaking with colleagues. And then you can figure out different aspects of the formulation of the nanoparticle in this case, and how that could relate, for example, to process parameters, and the final CQAs, the quality attributes on the output. On the right-hand side, we just listed some of the material attributes as an example, and some process parameters that we've looked at in the past. And then how this impacts or what this leads to the quality attributes, for example, particle size, polydispersity, and then having a better understanding further would be, what are the true biological effects of these quality attributes. So the particles that we've been able to produce using the technology are different types of liposomes, for example, lipid nanoparticles, polymeric micelles. So these are just some of the examples. We can also make emulsions and suspensions. So the platform that we've developed is really has broad use in the pharmaceutical industry. So how did we get to where we are today? And that's the idea into commercial opportunity here. In 2014, we started basically with just some small little valves and containers and air pressure, a pressurized container, and we were pumping liquid and forming particles. And then from there, we moved on to taking over the entire bench and really understanding or getting a better understanding of the process. For example, different stages that may include some heating, some different type of mixing, and then really trying to look at from where you produce the particle or the synthesis aspect of the particle all the way down, all the downstream processing included, to what would be drug substance and the potential fill finish, which leads us to in 2022, we actually have developed our full skid, which has all the downstream processing built in. So this skid here can produce the nanoparticle, it can concentrate, remove residual solvent, do buffer exchange, loads the API. In this case, this one we're doing some testing for liposomal doxorubicin, and and then we concentrate, purify at the ends, and all along the way we have process and local technology built in, so we can monitor and also have feedback mechanisms to control the entire process. So this is really getting to advanced manufacturing, leaning more towards Pharma 4.0 approach, and this we believe is really going to be the future of manufacturing. So some of the current nanoparticle, or on the current nanoparticle processing skid, just some of the overall features is that really has a broad range of applications. We have a fully integrated, adaptable approach, robust control with multiple integrated sensors and PAT. It's a continuous manufacturing approach, so it's a closed system going from beginning to end. It has rather high throughput, so we can go well over 1 liter per minute on the output, and then modular, low energy design. Now, just looking at the commercialization timeline here. So at the University of Connecticut, we started around 2014, 2015. We were granted a patent, or we filed our patent, in 2015, and we were awarded our first award through the FDA for about $500,000. And then over the years, from 2015 to 2019, we applied for different funding mechanisms within the FDA and continued to file patents. And then in 2019, we founded Diant Pharma, and that's our company that we're at this time equipment manufacturer, and having dual roles at the company and also at the university for a short period of time there, 2020 and 2021. And then on the Diant Pharma side, we had our first system sale in 2020, and then multiple system sales this past year in 2021. So all along the way, we were able to basically take advantage of the university, what they offer, their mechanisms in place. Also various awards for some of the development on the system. And so then just ending with the challenges along the way and the help. So clearly there were technology challenges, development cycle. We started this back in 2015, and then we started the company in 2019. So there was some time that was used, really for the development, the understanding the fundamentals, IP generations. This is where the university really came in. On the bottom of the slide here, I show one of the technology commercialization services at the university that helps with the IP generation, and then also they have some programs, Accelerate Yukon, which is a NSF I-Corps program that helps entrepreneurs, and then the Office of the Vice President of Research. They have various R&D initiatives, entrepreneur courses that are offered. So all this coming together really helped us start to commercialize this technology. Outside of the university, we then established the company, brand, started building a team, established some business development, and started to generate additional IP. Currently, we're also in the fundraising process. So really, going from grant writing to product sales and trying to license technology and sell product this was basically enabled through the various awards from the FDA, and the great spirit of entrepreneurship or entrepreneurial efforts and at the university has really paved the way and allowed us to commercialize this technology. So I'd just like to also make note of the collaboration and the support through the FDA on the various grants that are noted here, and I'd like to thank everyone for their time, and I look forward to today's workshop.
Thank you, Xiaoming and Tony. Appreciate your perspective on your collaboration for regulatory science. Just to remind our participants, we are taking questions at the end of this session. There are 2 more speakers. So please add your Q&A into the Q&A box, and please demarcate which speaker your question is for. And with that, I would like to invite Drs. Angeloni and Afshari for our next presentation.
Thank you. And if you can release the screen share. Thank you. Good morning. Good morning. Thank you for the opportunity of being here. I'm Leonardo Angeloni. I'm Deputy Director of the Office of Translational Initiatives and Program Innovation within the National Institute on Drug Abuse. And today, for this presentation, I'm participating also as my former hat as a FDA scientist, worked for over 20 years on safety of patients undergoing magnetic resonance imaging. And I'm pleased to be joined today by Dr. Ayman Afshari, who is a senior principal engineer from the Synthesis Johnson & Johnson, and also serves as the vice chair of the ASME verification and validation VVQ for the standard group. Just a brief background. Of course, we can talk for hours about magnetic resonance imaging, but wanted to share with you some of the key points that may guide our conversation today. And especially so when it comes to MRI, and we look at MRI safety of patients, MRI is a 3D detailed anatomical images provided, and there are three main components of an MR system, which is the scanner, the static magnetic field, the gradient coils, and the radio frequency coil. And each of them pose unique safety hazards for patients, especially patients with implants, external devices, and accessory medical devices. Of course, MRI remains to be overall a very safe device, just to make sure. And it is possible for patients with implanted device to undergo safely MRI under certain conditions. One of the aspects that needs to be controlled and studied is safety with respect to radio frequency field that are elicited by the MRI to have images. And this brings to labeling. So these conditions and constraints that allow patients to undergo safely MRI are categorized generally by three main labels. An implant is either MR safe, so patients can undergo MRI under all conditions. It's MR unsafe. Essentially, it is an item that poses unacceptable risk and patients cannot undergo MRI. And then the most, I think, interesting for today is MR conditional. So patients that have this device or implanted device can undergo MRI under specific conditions that needs to be studied, evaluated, and then they're typically added in the labeling of the device. The heating of devices and tissue around devices is quite complex. It's not straightforward. It's not linearly related to the dimension of the device. It's not the shortest or the longest device. There are aspects of resonance lengths, and there are being studies that frankly go back over 45, 50 years in terms of interaction of radio frequency fields with human body. There have been papers published by the Office of Science and Engineering Laboratories, Dr. Kynes, Bassen, and the Laboratory of Electromagnetic Fields. And the study of the effect of radio frequency exposure goes back and actually takes advantage of the plethora of studies that have been done back then, starting with interaction with microwave ovens and with cell phones in the 80s, 90s, and early 2000s. And then a lot of the people that participated to that actually started to also be interested in the effect of RF heating with MRI in the early 2000s, and then go on in the 2010s and on. One important example of this is that was part of the case study that we're discussing today is multi-configuration metallic implants. So imagine a patient that may have an orthopedic implant, and the implant may be composed of several screws. As I mentioned earlier, when it comes to conditions surrounded the plant. Essentially, radio frequency field interact with the metal of the implant and the receding surrounding this. It's not the longest, it's not the shortest that will give the worst case scenario. So when you combine several screws and several plates, here now you start to have millions and possible combinations, quite complex. So how you studied that? The original case was to use maybe phantom gel to try to understand the configuration, but obviously this becomes quite impossible if in just doing experimental studies. And this was also based in benchtop information, the old ASTM standard. This about around, as I mentioned, 2010 or earlier. A lot of work is being done with the community to introduce new tools and specifically computational modeling tools. And these tools were proposed as a complement, not to substitute existing approaches, but really computational models, just like animal models, bench testing models, gel phantom, and human clinical trials, which are also models of reality of what happens in a real case scenario, were all considered as complementary tools that really allow to study this quite complex situation, and ultimately allowed many patients with implants to safely undergo MRI and take the benefits of this great imaging tool. I'll pass now to Dr. Afshari to continue. Payman, you want to share on your screen?
Sure, as soon as, yeah, okay.
And once we do that, really I'm emphasizing that it was a collaboration of many on the FDA side, the Office of Regulatory Science, the scientists from academia and scientists from industry.
Thank you, Leonardo. Yeah, so from this point is basically my journey as on this path of getting that clarification of the electromagnetic. We had the heating. We had human models. There was no trust guidance. How do you trust the model once you develop a model? And there was no methodology. How we go about this finding a needle in a haystack of billions of combinations. And there was no reporting guidance also. Once you do that, how do you report the reports? And the acceptance criteria was not fully understood, at least from my perspective. So the road starts, curious enough, in 2012, I joined ASME VNB 40, which I think Tina was at that time the co-chair, vice chair as well. And one of the things about the objective of the standard was to develop a standard that you can use to assess credibility of models, computational model through verification and validation, so that you can see some activities there could answer some of these questions. And then, the answering to the original question was on the shelf while I was going through this. In 2014 or 13-14 timeframe, one of our homework problems, we had to review paper. And actually, it was one of the papers that was kind of brought some light into why this change of paradigm is happening. At least for me, again, is the uncertainty about testing, and the large number of possible configurations that should be considered for patients to be scanned. And so in 2015, the first thing happened was we did the framework, VNB 40 framework was somewhat established, and we were doing a test case. And we did a test case. We submitted a small project, and it went through the FDA, and it got cleared. So the no precedence was cleared. So there was precedence of using models in regulatory submission. And at the same year, we participated. We partnered with our software vendor, and we developed our multiphysics model. And a lot happened in 2016. The first thing was the reporting of computational modeling studies in medical devices. That guidance document was released 2016. So the no reporting guidance was cleared. And a methodology was introduced on the assessment of radio frequency induced heating under MRI environment for multi-configuration passive medical devices, exactly what we was missing. So by 2016, we had the multi-physics model, we had methodology developed, we have the reporting guidance, and we had some semblance of how the VMB is supposed to look like, verification, validation. And at the same time, there was a community effort, scientific community effort to develop updated standards to reflect the changes. And I think, and Leonardo was leading that for the ASTM F282. And the publication came, and one was the effort from the interlab studies from the same group of F282 that use models, the F282 task group, and again with many participants that use the model and simulation to see if consistency and if there's a good practice can be identified. And at the same year, there was Medical Device Innovation Consortium. There's a working group on the MRI RF heating. Again, Leonardo was leading that effort as well. And I think that was, for me, very, very informative. I learned a lot about the scientific community. And I think what came out of that was the acceptance criteria was slowly getting clear for me on the the dosimetry method that's being used to determine what's the safe level of heating for tissue, patient tissue. And in 2018, finally the standard was the ASME VMB40 standard was completely released, and so all devices got cleared. And we went through the methodology. We followed all the guidance documents by then, and we got cleared. So it's practical, and you can see the standards, the guidance documents. There's a lot of communities that contribute to that happening, to be completed. So it really takes a village. If you look at this, I'm reflecting back on all the stakeholders. You can see the scientific institution, the national labs, NASA, IDIS, the regulatory side, the CDRH contribution, the industry, the device, the software, the consulting, the OEMs, and the academia. We all contributed to through publication. All this collaboration happened through publications was necessary to develop consensus, which led to development of methodologies, the tools, medical device development tools, the standards and guidance documents. And that journey for me was, I met a lot of other scientists, and it brought a lot of clarity. And it makes this paradigm shift possible. So thank you very much.
Thank you. And if I can also re-emphasize, wanted to re-emphasize a message that as a former FDA, always encourage, and now as a NIH program office, always encourage our companies, whenever there is a new problem, it's really as an effort of collaboration also with the FDA, early approach to the problem, especially when there are new tools, new devices, when there is no standard. It really is very important to engage scientists within the FDA as well, the regulatory scientists, and together try to come up with what is the best approach, because ultimately we want devices to get safely and safe and effective device to get to patients, and to make sure that patients have access to these devices. It really is, this is one of the case studies where the entire community worked throughout the years. Thank you.
Thanks, Leonardo. Thanks, Payman. I really appreciate the emphasis here at the end on the collaborative community. For our participants, please add your questions to the Q&A box. After our next speaker, Dr. Kasia Shumakov. We'll have a chance to convene all the presenters from this morning and have a conversation to address your questions and see about the lessons learned. So with that, Kostya?
Yes, thank you, Tina. Let me share my screen. Okay. Oops. Hold on 1 second. All right. So I think this morning you already heard from Tina what the FDA defines as regulatory science. And I just want to emphasize that first, that of course it complements our mission, regulatory review of different submissions. It also allows us to proactively address some issues, challenges that are in front of the industry and agency. And what's I think it's also very important, it allows for global harmonization, because all results of our regulatory science that is done at the agency are in the public sector. So they not only benefit one company or one line of products, but the entire industry. But of course, the challenge is that how do we implement what is happening in our labs, our development that is taking place at the agency, and how do we bring it to industry and make it happen? Because we cannot just dictate to particular companies what they should or should not do. I mean, of course, there are regulations, but we cannot force them, especially those companies that produce licensed products. Since the marketplace is now global, regulations issued in one country cannot be applied to in other countries. So there must be some global mechanism how we introduce our results. So I will give you a case study of oral polio vaccine, which is of course very far from biomaterials or any issues that are of interest to the participants. But I think there are some important lessons that can be learned from what we've done. And I will give you two examples. So the vaccine actually is a live attenuated virus. So it means that's a virus that has been modified by passaging and some other manipulations. So it no longer causes disease, but can replicate in the body of a vaccine recipient and induces very robust immune response. It was introduced in the mid-20th century, and it resulted in a dramatic reduction of the incidence of poliomyelitis from several 100,000 cases a year when this was introduced to just less than 100 cases last year. So it's a dramatic success that actually led some to propose that the disease could be eradicated completely. So that's a global program on polio eradication is taking place as we speak. But the challenge of this vaccine is that, like all living organisms, it can mutate and change, revert back to virulence. So as a result, rarely, maybe 1 out of 3 million vaccine doses, it can cause paralytic adverse reactions in vaccine recipients just because virus can mutate and regain virulence. Another challenge is that this virus, mutated virus, can be transmissible. So it essentially it's a runaway vaccine. Vaccine virus becomes fully virulent and indistinguishable in pathogenic properties from wild viruses. So this is called circulating vaccine-derived polioviruses. They are now represent the majority of all cases of poliomyelitis. And this is a significant challenge. And what I will be telling you in the next few minutes is exactly to address this challenge. But it was realized early on that this vaccine can be used only after rigorous control. So from the very beginning of this vaccine, every single batch of vaccine was tested in monkey neurovirulence test. So just to release one lot of trivalent vaccine, we needed to kill 200 monkeys. So they were first inoculated intraspinally, observed for 17 days. Then all the monkeys were sacrificed, and histology evaluation was performed on the different parts of CNS. And then it was compared to the lesions that are caused by a reference vaccine. So it was extremely laborious, expensive, and inhumane test. So, but in the early 1980s, there's been a significant progress in elucidation of the molecular biology of poliovirus. Here you see the genomic structure of the virus, and I will not really dwell on this too much, except for I will tell you that on the 5' end there is a very long untranslated region that includes internal ribosome entry-site, where the protein synthesis begins, and there's specifically the one hairpin structure that is responsible for neurovirulence. It's been discovered in the 1980s that in all 3 serotypes of this virus, mutations that stabilize the stem loop, they result in higher virulence. And mutations that destabilize the structure of this hairpin, they tend to make virus less virulent and more attenuated. So that's what we thought. Could we simply, instead of looking for neurovirulence at the level of neurovirulence by inoculating monkeys, can we just simply look at the mutations that may take place in the in this domain? So, and then we developed this what we call mutant analysis by PCR and restriction enzyme cleavage that allowed us to quantify the amount of mutants in the batch of vaccine that was under consideration. So essentially, we amplified a short region of this genome and then digest it with restriction enzyme. And then you see the separation of bands in acrylamide gel of radioactively labeled DNA fragments. And by intensity of this band, we could judge on the amount of the mutant virus that was present in the vaccine batch. And to our surprise, we found that there was a remarkable correlation between the content of this particular mutant mutation and histological lesion score in monkeys. And here you see that green dots represent the vaccine batches that passed monkey test, and the red dots represent batches that failed monkey test. And this yellow dot is the US National Neurovirulence Reference. So it was a remarkable correlation. So that allowed us to propose that we, instead of testing vaccine batches in monkeys, we can simply do this molecular biology procedure and tell whether the vaccine will fail or pass monkey test. So, but the challenge was, how do we do this? How do we force or actually make a vaccine manufacturer use this technique? Of course, they would be interested perhaps to save some cost of monkey test, but still it was challenging. So we decided to use this mechanism. We contacted the World Health Organization and proposed that we organize a collaborative study with participation of vaccine manufacturers, national control authorities, of regulatory authorities from other countries. And from roughly 1992 to 1995, we conducted this study. It was a really collaborative effort where everybody got used to using this test, and they actually could personally, learn about how it performs, and they realized that it's really a very valuable tool for lot release. Then in 1996, the Expert Committee on Biological Standardization, which is a WHO committee, they recommended that MAPRIC, this test, would be introduced as a preferred in vitro lot release test. Then, of course, we were very encouraged, and we conducted similar collaborative study for 2 other serotypes of polio vaccine. And roughly at the turn of the century, ECBS endorsed MAPRIC as a lot release for all 3 serotypes. And since then, it became a kind of routine test, and every single vaccine manufacturer started using it. I think it's a very nice example of how the regulatory science that we do at the agency can be implemented into the regulatory practice. So we've done it in collaboration also with our sister agency in the UK, NIBSC, National Institute of Biological Standardization and Control. So it was basically a kind of collective effort. But we were not fully satisfied with this because, of course, the genome of the virus is made of 7,500 nucleotide bases, and we were looking only just one. So what if there are some changes happening somewhere else? So that's why when the NGS, next generation sequencing technology, became available, we decided to take to look whether it can be applied here. So you can see, for instance, that there's a content of mutants in the entire genome of poliovirus. So we sequenced it. At the time, we were thrilled to sequence it 10,000 times. So it was a massively parallel sequencing that allowed us to quantify mutants in the entire genome of the virus. So here is the profile of mutations of a failed vaccine batch. And this is a profile of mutations of a good test that passed monkey neurovirulence test. So you can see that there is a clearly, there are much more heterogeneity, sequence heterogeneity in the bad vaccine. So again, we decided that we will use the same mechanism and started a collaborative study, which is still ongoing under the auspices of WHO with the help of PATH, another nonprofit organization, and it's still going on. But our expertise in this next generation sequencing and actually our leadership in the field allowed us to participate in yet another initiative that was organized by the Bill and Melinda Gates Foundation, because essentially what they wanted to do is to develop a new next generation of polio vaccine itself, based on the detailed knowledge of its structure and biology and molecular biology. It was proposed that the viral genome could be modified so that it will no longer can revert to virulence. So basically to stabilize, genetically stabilize the genome by targeted genetic manipulations. So the consortium of several labs was put together by the Gates Foundation, and it included NIBSC and UCSF. And then there was a group of scientists from the US government, CDC, Olin Q, Karen Burns, and they've invited us at the FDA to participate in this effort, again, based on our leadership in this NGS field. And also there was another participant from academia, Dr. Wimber from Stony Brook University. So it was about 10 years ago. And so there were several candidate strains developed. Here's one that actually came to the finish line. So first, of course, this domain 5 that I've already told you, it was modified in a way that it can no longer mutate to stabilize its structure. So all GU pairs were replaced by AU pairs, so that you can only change it to make it stronger by 2 mutations, not just one. So it would dramatically stabilize the structure in terms of its genetic stability. Then this cis-acting replicative element that is essential for replication of the virus was inactivated where it was located initially, and it was transplanted into this part of the genome so that recombination cannot get rid of this stabilized structure. Then finally, there were 2 mutations introduced into the polymerase gene that made the polymerase much more accurate. The fidelity, it's called hi-fi mutations that make virus more accurate in copying its genome, so that mutations can no longer, they occur with much lower frequency, as well as mutations that actually block recombination. So as a result, this vaccine was tested first in cell culture, and here are results that we obtained with conventional Sabin virus. And you see that during passaging, there's a dramatic accumulation of mutations, while this novel OPV strain was much more stable. So at least until passage 7, there was not much going on in the genome in terms of mutations. So as a result, there was a clinical study conducted, and this was facilitated by PATH. And at the end of 2020, the vaccine was actually introduced into practice under the emergency use listing by the World Health Organization. It's a kind of similar to what we call emergency use authorization in this country. So as we speak, it's already 260 million doses were distributed worldwide. And the results of this massive trial actually demonstrated that the safety of this vaccine is by far superior to the original vaccine that was used since the last century. In conclusion, I just want to basically tell you once again that, of course, we are uniquely positioned at the agency to conduct this type of studies because, I mean, we are actually exposed to a lot of new developments in the industry. So we are aware of the challenges of biomedical industry. But of course, it's very difficult in terms of, trying to maintain impartiality and try to avoid any conflict of interest. So that could jeopardize the authority with which FDA regulates industry. But the way around this, basically the pathway for the implementation of the results of our regulatory science is the collaboration with the international organizations such as WHO, nonprofit organizations, sister agencies in other countries, and as of course also other US government agencies that can actually provide some kind of firewall between FDA and the industry. And I think that this is something, this paradigm could perhaps be used in fields that are closer to your interests. And I think that is all I wanted to tell you this morning.
Great. Thank you so much, Dr. Chimukov. I appreciate that. Let's see if we have our panelists, our speakers from this morning. Just to remind our attendees, they can add their questions to the Q&A. I believe Dr. Zhang is moderating questions in the audience. For those of us online, unfortunately, we can't see you. Will the speakers from this morning please turn on their cameras? I believe that will allow folks in the audience to see you on the big screen. We do have a question this morning for Dr. Kostya. Has the research resulted in changes to FDA guidance or national or international standards? Maybe you can tag team with Xiaoming on that one.
Yeah, probably better answered by Xiaoming. I can say that there's a lot of information that from these awards that from meetings that we supply to the FDA. And I think Xiaoming will be better suited to answer what is done with that information.
Yeah, thank you. So for the guidance, typically it's a long journey before FDA can issue a guidance, and typically the guidance reflect FDA's current thinking and understanding on a particular topic and area. So in this case, what the example we shared this morning was an innovation in the area of manufacturing, which I would consider is still early phase in the development, where we're pushing the boundary of what's new and we're gather understanding. Now the hope is that we will take that understanding, internalize within the FDA, and help with the reviewer in terms of training. And in that sense, maybe the follow-on steps, once we gathered enough understanding, then we will hope to translate that into a guidance document to help the industry understand better about the regulatory expectation for a particular area of advancement. In this case, the continuous manufacturing. But I think maybe flip the other way in terms of the question. I think what this research example showcases is actually how the guidance which FDA has already published in the area of innovation, continuous manufacturing, quality by design, how actually that can be translated or actually demonstrated through an example of research to develop this technology and to show the advantage of those innovation in the area of manufacturing. So I think in short, it's going to take time, but we hope that's the direction we may be moving forward to.
Yeah, thank you so much. I'm going to just pause for a second just to let our audience members know. At this time, we were supposed to start our break. What we're going to do is we'll continue for another 5 or 6 minutes with a few questions from our panelists, and then we'll take a quick break. We'll have a stretch break for those on Zoom. In the room, you all can get some more coffee, and then we'll join back at 9:30 to kickstart the second session. Tony, did you have additional comment to add from Dr. Zhu?
Tina,
Yes.
we
Dr.
have
Zhang.
some question in the room.
Awesome.
Yeah, no additional comments.
Dr. Zhang, is there a microphone in the room, or will you relay the question? Dr. Zhang, if you're speaking, we can't hear you. Oh.
Yeah, I'm gonna. You do this. Can you guys hear me?
There's a little bit of feedback, so give it a try.
Hi, I have a question for Dr. Chumikov.
Great,
Yes.
please go ahead.
Can you still hear me? Okay, our feedback's
Yeah.
up. Okay, so I'm really curious how you were able to go from the development with the Gates Foundation of the NOPV2 to the 260 million doses disseminated worldwide. Like, what was the transition from that? Was that through the FDA more so, or through the Gates Foundation? Like, how did that translation happen?
Yes, it was completely through the Gates Foundation.
Now we can't hear.
Can you hear me?
This doesn't show, but you still have connection.
Yeah.
We can hear you on
You
the
guys
Zoom,
hear
but
the
I'm not sure
question?
if they can hear you in the room.
Tina,
We can hear you.
can you
Yeah.
guys answer the question?
Yes.
Yes, he
right.
was.
Yes. Can you hear me? I
Yes.
hope.
Yes.
Yes. All right. Great. Yeah. So definitely this project, this whole consortium was coordinated and funded by the Gates Foundation, and the Gates Foundation actually contracted PATH. You know, this is a nonprofit organization working in the public health field, who also manufactured this vaccine at the Indonesian company Biopharma. And there was a lot of different organizations involved. And our role was only to provide our expertise and conduct some studies with the genetic stability. And also what's important, since, we're the regulatory agency, and even though this vaccine is not licensed and will never be licensed in this country, so there was always kind of concern that it could basically create a perception of a conflict of interest. So we worked in collaboration with the CDC. CDC is not a regulatory agency, so we worked on an interagency agreement. So we essentially helped CDC to complete their international public health mission. So that was a kind of firewall that we built to prevent any concerns about the impartiality of the agency.
So complicated. It's really interesting.
It is, but we have to be very careful, as you understand.
Yeah.
We have other. Sorry about that.
Is there another question from the room, Dr. Zhang?
Yeah, looks no. So we're gonna take a short break.
It's a question online.
Okay. Yeah,
Come
there
back
was
at
one question,
9:30.
one more question
One
from
more question.
online, and then we can take a quick break. And I'd like to probably address this to Payman and to Leonardo about expecting the future of in silico standardized testing to reduce, refine, and replace traditional bench in vivo testing. I think you guys demonstrated an example of that. I would say, what do you think your expectations are to make that happen?
It's already happening. Certainly, they are reducing the amount of testing done. They are refining. Regarding replacement, I think it's very important to, as I mentioned, and I keep emphasizing, computational model is complementary to existing tests. I think we will never just have models because of course, you still need to have validation, experimental validation. But as far as reducing refinement, that's already happening.
Yeah, Payman, anything from the industry side you want to add to that?
No, I think Leonardo made covered it pretty well. But it's evolving. It's definitely evolving. Our knowledge is, through these collaboration environments, right? Our knowledge is expanding. So we have a set of tools, but it's like a hammer looking for a nail sometimes. But the new, all the AI, ML, data science, data that's coming, with the different view of the models and their application. So it's an evolving environment, both for, I would say, for the scientific community, for the applications community, and the regulatory community. This is definitely a rapidly changing environment.
Emphasize that CDRH recently, the past 2 years, there are 2 new medical device development tools specific for MRI, and this I think is a great example of the work that continues to be done also by OSL in making these tools regulatory level.
That's right. Well, thank you all so much. Let's do a virtual round of applause for our speakers online and those of you in the room, and we'll take a short break and we'll be back at 9:30.
Thank you.
So for those of you who joined a little bit later this morning, I'm Tina Morrison. I'm the Director of the Office of Regulatory Science and Innovation in the Office of the Chief Scientist at the FDA. FDA's mission is to protect in public health, and that's a part of our mission that I think most people are really familiar with. Another part of our mission is to advance public health by helping to speed innovations. And legislation that took place in 2007 allowed for the agency to create the Office of the Chief Scientist to work at the agency level to increase scientific coordination across FDA in a way to reduce organizational silos. So OCS's goal was really to help forge across agency effort in vital areas of regulatory science, which I'll talk about in a few moments. We have 7 offices in the Office of the Chief Scientist. One of them is the Office of Regulatory Science and Innovation, which I'll tell you about today. Part of the Chief Scientist engagement activities are to conduct scientific research and foster innovation and collaboration in support of FDA's centers. I showed you this slide this morning in my opening remarks, and just to orient all of us on the same page, we're talking today about regulatory science, which is the science of developing tools, standards, and approaches to support the assessment and evaluation of FDA-regulated products. And something that I think folks who haven't had a chance to work with the FDA before don't realize that FDA is instrumental in advancing regulatory science because we actually conduct science. Each of the product centers at the FDA have their own regulatory science facilities. Some of our centers, like the Center for Biologics and Evaluation Research, who you heard from Dr. Chumakov this morning, those researchers actually wear 2 hats. They are both principal investigators on regulatory science research, and they support the regulatory process. In the Office of Regulatory Affairs and the Office of the Chief Scientist, we have several offices that support FDA's regulatory science mission. There are several thousand scientists at FDA. One of the central places I want to think about today's conversation, when we started this discussion, we wanted to think about life cycle approaches, if you will, to regulatory science. And FDA's important role is thinking about identifying knowledge gaps, executing scientific research programs. When we get that scientific knowledge, we have to communicate that back into the scientific community, both inside FDA and outside through publications, presentations, workshops, etc. We then create or update regulatory guidance, standards, methods, and capabilities. And then we harness that regulatory science knowledge by way of hopefully informing our regulatory processes, but also making sure we share that information with our stakeholders. I want to take a minute just to talk a little bit about regulatory science outputs to kind of make a connection between the presentations that you heard in the first session and where we're headed with the second session later this morning. So some of our really critical regulatory science output are tools, standards, and guidances. And the reason why these are so important is that these documents, this information can then inform the regulatory process. So guidance documents are our interpretation of the regulations. It's really FDA's current thinking on a particular topic, but they do not create or confer any rights for any person to use, and they're not binding. So when you see a guidance document, you heard Dr. Angeloni this morning talk a bit about a couple of guidance documents that were in the modeling and simulation domain. What's really important about FDA's perspective on regulatory science that's put in a guidance document is that once we put our materials together, this is a place where regulatory science happens at the FDA, where FDA is instrumental in putting forth this guidance document. We do put those materials out in the draft format, like we did this year on one for assessing credibility of computational models. And then FDA can then use that final guidance in regulatory processes. Another area that's really critical to our engagement in the community is through consensus standards. And standards are really important because they allow the community to come together on methodology that folks are using in the different aspects of the community to create what's called a standardized approach through consensus building. What you heard from our presenters this morning is that both guidance documents and consensus standards development can take a lot of time. Bringing stakeholders together, deliberating on methodologies, processes, ensuring things are repeatable and can be used by all stakeholders, both industry and FDA, and of course our academic colleagues working on the cutting edge of innovation. It's really important that we take the time to identify those critical areas, needs, and gaps. And there are many different types of standards. You heard about standards that are helped to elucidate a particular type of methodology. There are also reference materials or scientific protocols that can be used in the standards development process. And then there are tools. And tools are methods or materials or measures that can be used to support product development or product evaluation. Two of our centers, the Center for Drug Evaluation and Research and the Center for Devices, they both have extensive tool programs geared towards developing tools that support both the industry with their regulatory submissions and both FDA with their regulatory review. How do we identify which areas we should work in? Just last year, FDA updated its regulatory science focus areas with a report identifying both to the community outside FDA and inside FDA on what are the target areas that need continued investment. This report was published 1 decade after our original regulatory science strategic plan. This current report highlights several different areas that we would like our stakeholders to continue doing important regulatory science work. One thing I want to add is that I mentioned a few moments ago that each of our centers, our product centers, have their own regulatory science laboratories. They do their regulatory science work in those particular targeted areas. What we're trying to do in the Office of the Chief Scientist and by putting together a comprehensive report like this is to really identify areas that are cross-cutting to the agency in which we can help take a leadership role. These focus areas of regulatory science are categorized into 4 buckets. What we're calling our strategic initiatives, public health response and preparedness, increasing choice and competition through innovation, unleashing the power of data, and empowering patients and consumers. So you can find this report at fda.gov/FARS. This is a quick snapshot. In the interest of time, I'm not going to go through the details of the FARS report, but I do encourage you to go to our website and take a look. I'll also encourage you to take a look at a blog post that we published last spring, which highlighted a call to action for our stakeholders. It's really important that when we think about these focus areas of regulatory science, we're not just thinking about those particular areas as related to, say, one product area like medical devices or in drugs, but that we also think cross-cutting about our specific populations in the area of pediatrics research, looking at racial and ethnic minorities, women, patients with rare diseases, and different diverse groups. We also encourage input from our stakeholders, and in fact, that's one of the reasons we're here today having this roundtable, is to think about ways in which we can get input from the scientific community outside of the agency to give us their expertise and perspective on other target areas that need continued investment. So I encourage you to review the report and provide us with your feedback at fars@fda.hhs.gov. And we'll continue with further community engagement activities like this one. I'll take a few moments to introduce you to the Office of Regulatory Science and Innovation, where our mission is to accelerate innovation through creative collaborations that harness the best science. And our mission is to provide excellence and innovation in strategic leadership and coordination, infrastructure development, to make sure FDA continues to have a strong regulatory science foundation. And we do that through several different mechanisms: scientific engagement and coordination, through an extramural contracts mechanism, through cooperative agreement grants with our academic colleagues, and also intramurally with the FDA scientists working across the agency. I'm going to briefly touch base on a few key areas that I want to make you aware of with respect to regulatory science, one of which is our scientific working groups. We have 12 cross-agency working groups in cutting-edge scientific areas where we bring together representatives from each of the FDA product centers, along with our associated offices in the Office of the Chief Scientist and the Office of the Commissioner. So each of these working groups has anywhere from 12 to 14 members that on their leadership circle that are guiding the direction of regulatory science work happening in any of these scientific areas, such as toxicology, alternative methods to animal studies, advanced manufacturing, artificial intelligence. So we are active in our areas of engagement through these scientific working groups. We also have the CERSI program, which is our Centers of Excellence in Regulatory Science and Innovation. This is where we bring together expertise and research from academic institutions working on the cutting edge of regulatory science. They work collaboratively with our FDA regulatory and scientific expertise to advance regulatory science. We host training, but the bulk of the work that we do through this program is regulatory science research. Currently, we have over 100 projects through the CERSI program, and our current cooperative agreement grant cycle ends this coming fiscal year, and next year we will be launching our new Centers of Excellence in Regulatory Science. So if you're at one of those academic institutions that's interested in joining, our next request for applications will be later this summer. One area that's really critical, you heard from Dr. Kasha this morning, we have our Advancing Regulatory Science Broad Agency Announcement Program, which is our extramural contracts program, where we spur scientific innovation in different areas to address gaps in needs, both in expertise and infrastructure. And we do that by targeting our colleagues outside the agency. Whether you're at a laboratory facility, at an academic institution, you can apply for one of these extramural grants in any of these target areas listed below. And we have a very rigorous review process by which we review the quad charts and applications and proposals with teams of experts from all across the agency. So I encourage you to take a look at this as an opportunity to kickstart your innovative work that's happening at your institution. Before I end, I just want to reorient us on this concept of a regulatory science lifecycle and how we identify knowledge gaps and kickstart regulatory science work. From my perspective, it's really critical that we get engagement from our stakeholders in the ecosystem to maximize the benefit of regulatory science, not just for FDA, but also for its stakeholders. So I look forward to input from all of you on how best we can do that. When we share our slides after this conference, I've provided several links to help you navigate all the different programs I've introduced you to today. So with that, I'm going to pause and then pass the podium over to my colleague, Dr. Scott Steele.
Thank you, Tina. I will pull up my slides here. Can you see those working?
Yes, thank you.
Great. Thank you, Tina. And thanks again for the invitation to participate in the discussion. I'll try to make up a few minutes on my presentation. So really appreciate the opportunity to join you. And as Tina mentioned, I'm with the University of Rochester and our Clinical and Translational Science Institute, and also involved in some of the Clinical and Translational Science Award hub institutions activities. So we have a special interest group focused on regulatory science. So I'll touch on some of the activities we're involved in to really try to integrate some of the regulatory science themes you've already heard about into some of our translational science programs, both for research, but also to particularly advance some of our training initiatives. So Tina already reviewed very nicely some of the definitions. So I'll just mention this briefly, since this helps, particularly for our CTSA network, the priorities and the definitions that have been set out by NCATS, the National Center for Advancing Translational Science, that houses the CTSA program, and really viewing translation as a continuum, and thinking about the process from basic research all the way through translating to impact public health with a patient-centered focus, and thinking about the field of translational science, really looking at that entire process and feedback loops between different phases of translational the translational process, and really trying to improve the effectiveness and the efficiency of that process as a whole, and also in some of our translational research priorities, looking at individual steps throughout that process across the continuum. So in thinking about that, in the early development of translational research and as a field, a number of the CTSA institutions set out to help develop some competencies that could guide training and education in our programs. And I've shown these here, again, from some early work that could think about what are some of the core competencies around different scientific research questions, to designing and implementing research studies, to addressing some of the key needs around statistics and informatics and different computational approaches, to community and patient engagement and some of those key research collaborations and getting patient input early on in the development of a study, to scientific communication, both technically and with the public, to some of the multidisciplinary and team science elements of translational research. So those have served as a nice guide as we develop both formal and informal training programs for our translational science initiatives. And more recently, NCATS and others have really thought about some of the key characteristics of a translational science. This is from a 2019 publication, and it really aligns with those competencies and thinking about some of these core principles around communication and being both an expert in a particular scientific domain, but really recognizing the team science nature of both translational science and regulatory science, and thinking about that both as a field as we develop translational scientists and support their research. So switching to regulatory science, Tina just talked about the definition of regulatory science, which has really been helpful for us, and I think for the translational science community as we think about how to complement and integrate some of these core principles. And as was mentioned, the more recent focus areas of regulatory science, and I just highlighted a few in red that I think particularly complement well some of the translational research priorities that we've really focused on over the years across a number of institutions, from more certainly with COVID and other public health emergencies, to think about developing how to better develop and prepare for new devices, diagnostics, medical countermeasures, to substance use disorders such as opioids and other issues that are really of great public health need. To more precision medicine, pharmacogenomics approaches, looking at novel trial designs, adaptive trials, other types of trials that are being used. We've talked a little bit about regenerative medicine and tissue engineering and biomanufacturing already, and some of the scientific needs there, to the range of different medical device and drug development tools. Some that have been touched on around biomarker development, to modeling and simulation, both for PK/PD, but for dosing, for really even modeling clinical trials as a whole that could help guide our work. And of course, that relies on really a diverse and rigorous data sources and methods from AI and machine learning to use of digital health tools to capturing and taking advantage of real world data from the health records to claims to registry data, and then back to this patient-focused engagement and communication. And so this has been a really helpful guide in some of our research activities. And of course, different CTSAs and hub institutions have programs to support both that we've been piloting and experimenting with to support regulatory science research activities, as well as this core regulatory affairs component. So just briefly, in some of the regulatory science spaces, there's a number of different pilot programs to help seed research areas that are generally agnostic to specific disease types, but programs that may focus on use of digital health technologies or precision medicine approaches, CAR T types of therapies, to supporting new research methods and tools, whether you're integrating digital health technologies into a clinical study or using decentralized clinical trials, different methods for adaptive trial designs, providing novel biostatistics approaches, to again, back to training programs, whether that's informal degrees or integrating regulatory science components into our translational science certificates or pre-doctoral or postdoctoral programs. And entrepreneurship and innovation was mentioned in the prior session. And I know a number of us have also integrated entrepreneurship programs using the I-Core models and others to also integrate that innovation and technology development considerations into some of our regulatory science initiatives. And then on the regulatory affairs piece, which is of course a core element to ensure compliance with current regulations across the whole translational science spectrum, and generally there's regulatory support offices that provide that critical service, whether it's for GLP or GMP compliance, to human subject protection considerations, to looking at submissions of new drugs, new device applications, those interested in trying to take advantage of expanded access or other types of programs. And again, on the regulatory affairs piece, a range of training resources to help our investigators and clinical research coordinators with their studies. So I think a number of complementary programs, both to support the innovative regulatory science elements as well as the core regulatory affairs compliance. And so particularly thinking about regulatory science training, part of our special interest group and its predecessor, really as the regulatory science strategic plan was released in 2011, and some of the initial scientific priority areas, really felt there was a lot of similarities to the early days of translational science as a field and some of the needs it had. So some of the areas we've focused on is developing, I mentioned the translational science, clinical and translational research core competencies. We set out to develop a similar set of regulatory science core competencies that could help guide training and education, again, both formal and more informal training opportunities for regulatory science, and what that kind of knowledge, skills, and abilities would be helpful. That included different diverse training experiences across different sectors, as we've heard about a bit already, having that beyond the academic research experiencing, having at least some level of exposure of the work and needs for industry, as well as with regulatory agencies. And that includes developing some internships and fellowship opportunities. Partly also just increasing, as we're doing today, increasing awareness around regulatory science. We have researchers in diverse departments from biomedical engineering to pharmacology to neurosciences, whose work is really directly relevant to some of these regulatory science needs and may not be aware of that. And then a theme for us today as well, just the opportunities to partner across institutions, not only academic institutions, but particularly through industry, FDA, through these types of public-private partnerships that are really valuable. So as I mentioned, some of our early work was to really define a set of kind of core thematic areas and then specific competencies. And this was really a collaborative effort across different academic institutions, the CTSA hubs, working with Pharma Foundation, a range of other industry sectors, NIH, FDA, and others. And I won't go through them here, but these are kind of the core areas that we developed initially, certainly focused more on medical product development, both devices and drugs. There's been some interest to expanding that to some of the other areas, of course, that FDA regulates around food, veterinary products, and tobacco, and other areas. But we were very more focused on the medical product development spectrum. And then over the last few years, we've started to institutions have integrated that and utilized these competencies for certificates and other formal programs they've developed, but also thinking about where we can utilize case studies as a training module, internships, fellowships that can really help match certain competencies that we're trying to meet for different sectors. And then again, I think in many ways there's a lot of alignment in as we think about translational science and regulatory science. Certainly some areas that may fall outside of each, but in areas around thinking about the team science importance for both areas, the multidisciplinary nature, some of the key expertise in certain domains and those focus areas that we discussed, and really building on some critical analytical approaches and the need to communicate, again, both technical communication, but also with patients and broader sets of stakeholders. And so tying into the kind of the partnerships and how we can continue to support and align translational science and regulatory science, I think the CTSAs provide a network both for training and research. Tina mentioned the CERCEs and perhaps expanding CERCE programs that really have critical both research and training initiatives underway. And then through the BAAs that were mentioned, other types of demonstration projects. I think we'll hear from next from Klaus and the Critical Path, Reagan-Udall Foundation for NIH, and other groups where there might be opportunities to really leverage different research and training initiatives in a more holistic way. And so just to sum up, I think all of those partnerships can help both individual elements of translational research, but also the spectrum of translational science more broadly. And certainly there's a lot of activity in that area, but also I think opportunities for greater alignment between some of we have the focus areas of regulatory science, and thinking about FDA and NCATS and others where there's opportunities to more fully align some of those priorities. Thinking about a needs assessment, and on the training side, part of what our group now is starting to do a bit of a more formal needs assessment from industry, from some of the government agencies on what are some of the both current and on the horizon needs for regulatory science training, and where do they see gaps. And again, leveraging some of these networks, including around, I just mentioned the rare disease, other parts of NCATS initiatives around rare diseases, the Centers for Accelerating Innovation, REACH, and a range of other programs. So I look forward to continuing the discussion. Thank you.
Thank you so much, Scott. That was excellent. Great synergy between our talks. And I know that Dr. Romero will also provide another perspective. He's there in the room with you, Dr. Zhang, so I'm going to pause and turn off my camera and hope that helps facilitate getting Dr. Romero to the podium.
So come to this meeting and share perspective on regulatory science. We discussed how to best give a perspective on regulatory science that actually had a tangible application, so that it became something that could be more tangibly depicted. So I'm going to talk to you about an effort that went from integrating patient-level data, that's a challenge of its own, from trials from industry with observational studies and patient registry, so a collection of clinical trial data, observational studies, and real-world data. To then generate a series of actionable solutions that could transform, and that did indeed transform, the drug development process for an orphan condition holistic kidney disease, that's PKD. Okay, so the work that we did at the Critical Happiness Institute, we are an organization that provides the legal infrastructure, the scientific infrastructure, and then the regulatory infrastructure to generate a neutral environment so that industry and academics and regulators and other government agencies can come to this neutral environment that we provide, and they share information that they would not be able to share otherwise. Typically, that information takes the form of patient-level data from multiple sources. As of last Friday, our count is just under 500,000 individuals across clinical trials and observational studies and registries, and now electronic health records across a collection of 18 therapeutic areas. One of them is PKD, and that's the example that I'm going to show you here. The three things that you see on the slide, all anchored around the center on this abbreviation MIDD, Modern Informed Drug Development, came from the data that came from the different sources, and we have a rare disease data platform dedicated for these orphan and rare conditions. Then we have a PKD consortium, which brings the power of the collaboration from industry and academia, specifically about the disease. And then we have the Quantitative Medicine Program, which is a program that I helped start a number of years ago at the organization, that really takes the data and transforms the data into actionable solutions. What are those solutions? So the first solution was to take the data and run some biomarker models to generate some evidence that total kidney volume, which is an imaging marker, could actually be used to optimize patient selection for these kinds of trials. Okay, now the landscape before doing this work was that there was nothing really, there were no drugs specifically approved to have an impact in the disease. Okay, so we started with that integration of data, and we started analyzing the data and presenting and generating the evidence so that the FDA could reach a qualification decision. What is that? That is a level of a regulatory decision that is not about a product. It's not about a device, it's not about a drug, or a biologic. It is about a drug development tool. And total kidney volume was presented as a drug development tool for that kind of a decision. And what you read on the slide is the actual text of the qualification decision by the FDA. Right? So prognostic enrichment biomarker to select patients, okay. At risk of progressive decline, okay. For inclusion in trials, okay. That's great, but what does that really mean? What kind of evidence did we need to show to the FDA to reach that decision? It was essentially a model that looked at the progression of total kidney volume over time across all the data sets that we integrated, joined with a survival model that looked at how the TKD dynamics drove the time varying probability of individuals losing about a third of their renal function as expressed by eGFR. And we had to include a whole bunch of sources of variability in the analysis. And those were the ones that made it to the top. Okay, fantastic. So now we could offer industry a tool to optimize the selection of patients, because one of the challenges that they had was, how do we select patients that are really good patients in an orphan condition where recruitment is hard enough to begin with, but making sure that we select the patients that are really going to likely be good candidates for the kind of trial that we're looking to design. Okay, but then what? The tool itself can then be used to understand how you can put all those baseline characteristics and run simulations based on the model to then say, this is how I'm going to enrich my trial. And this is important because typically when you talk about a biomarker for selection of patients, the mind tends to go to, give me the threshold. What's the threshold that I need to pay attention to so that I put the patients in or not put the patients into the trial? Defining a one-size-fits-all threshold for a biomarker is not easy to do. Mathematically, it's easy to do, but you need a lot of data. Look at cholesterol. Decades upon decades of research, millions of data points, and we're still moving that threshold around. Think about total kidney volume when this was the first time this was done. So what is different about the kind of evidence that we presented and the kind of evidence that was generated is that the model itself treats total kidney volume as a complete continuous variable. And what industry can do is run the simulations to determine a trial-specific and drug-specific proposed cutoff point to select patients for their trial, for their drug. We're not making claims that we have a one-size-fits-all threshold. And that's what was really relevant for the discussion with the FDA and the EMA for the qualification of total kidney volume for patient selection. Okay, but then we continued to work and we started leveraging the same kind of evidence and the same kind of dialogue to eventually reach a point where the evidence was compelling for the FDA to conclude that total kidney volume was not just good to select patients, but it was also a reasonably likely surrogate endpoint for trials. And this is important because that means that that opens the door for accelerated approval. And that is exactly what was done for tolvaptan. One of the members of the PKD consortium had this asset. They leveraged all the work that we provided through the consortium, and they benefit from that by having an optimized set of designs of trials and then have a successful approval from the FDA for the first drug ever to be able to slow down or change the course of progression for PKD patients. That's fantastic. But that also led to a component of the pipeline growing. And now there's more than 20 drugs under evaluation at different stages for PKD. And the tools that were generated through this collaborative effort with a focus on actionable regulatory science were the transformative piece. Now, the work has not stopped because now sponsors are asking, okay, so this is fantastic, but the tools that are out there, the tools that have been generated are great for certain questions, but you see a lot of no's. Answers, or ask answers to the questions that are really being asked for drug development in PKD. There's a lot of questions about how you properly design a trial and properly execute a trial that the current tools don't necessarily provide. And that's why we have these consortia that pay attention to those needs and then start generating the tools that are needed. It's not like after we got the qualification, everything stopped, or after getting the designation of a reasonable surrogate endpoint, everything stops. That's another thing that we hear from our colleagues in industry. If I had a dime for every time I've heard the following statement, I would probably not be here. All our problems are solved through a surrogate endpoint. Well, you have a surrogate endpoint here, and you see that still you're having no as an answer to very relevant questions about trial design and execution. So what we're doing right now is developing a series of additional models that take the previous work, but enhance the work so that we can understand not just the longitudinal dynamics of TKD, but the longitudinal dynamics of TKD together with the longitudinal dynamics of EGFR, and then joining that not just to a percent loss of renal function, but to really understand the time varying probability of reaching end-stage renal disease. And what we have so far is the first component of the model. And what we're doing now is develop the other part and then put that into a package of a clinical trial simulation tool that will then be presented again as a drug development tool to FDA and EMA. Now, this is where things get a little bit interesting in terms of the regulatory lingo. FDA does not have a qualification pathway for these types of drug development tools like clinical trial simulators. They have another pathway called the Fit for Purpose Initiative. That's what FFP stands for. EMA qualifies anything and everything. Well, it is what it is. I'm not going to offer an opinion on one or the other. Each agency has its own set of procedures. Let me show you how things look like. So this is the model that is now the latest model that looks at both the longitudinal dynamics of EGFR over time and total kidney volume over time. But I do have a further update, which is this. So this is how the parameters derived from these models that you see here that describe the longitudinal behavior of EGFR and total kidney volume drive a time to event model for ESRD that helps you understand the time varying probability of hitting that kind of an endpoint. Look at the time scale. We're looking decades here for this one, thanks to the data we have. So this is again, kudos to the contributors. And look at the time scale here, also in years. But frankly, I mean, you could drive a trial for 1 or 2 years if you're evaluating a drug. You can't run trials for decades. Luckily, you have TKV and EGFR as metrics that you can use for trials of reasonable duration. But for ESRD, you need the projections based on sound data that you have to then be able to design trials that not only allow you to optimally evaluate the effect of drugs on TKV and EGFR, but also potentially evaluate what is going to happen with end-stage renal disease later. And so, yeah, a lot of sophisticated modeling, but what for? Well, so that we can start having yeses as answers to the questions that are tangible questions and practical questions from industry to design these trials. So everything that I showed you right now is an example of regulatory science. And regulatory science never stops. And what we're aiming to do is continue to expand the PKD consortium database, leverage the quantitative medicine program, and provide the solutions that are needed by the community, everything with a focus on actionable regulatory science. This is how we make it happen from the data to the solutions. This is the platform and the setup that we have for the rare disease data platform, which is the only way that we can get to this part of the slide. The actual drug development tools, we need to integrate a whole bunch of different data types and be able to integrate them in a way that actually can then support some sophisticated analytics. But nothing of what we do happens in a vacuum. That's why we have the consortium, that's why we have the collaborations that we have, so that we can get to the end at these set of solutions that really transform drug development. And so the consortium will continue to transform paradigms, and this is the example I have for you. Notice that I avoided showing any equations. I learned that for many years that equations can be scary, but I'll just leave you with one equation. Success is a function of time and people. So I thank you people here in the room and virtually to spend the time so that we can continue to be successful in transforming paradigms for drug development through the powers of regulatory science. That was that. With that, I'll conclude. Thank you.
Thank you so much, Dr. Romero. Let's see if we can turn off the slides and get the panelists up. I see Scott's ready. Guigan, I hope you can help manage if there are any questions in the room. We'll take about 10 minutes of questions, and then we'll have a 10-minute break, and then we'll resume for our panel at the end. I do have a couple questions as well for our speakers, but we do have a question from Dr. Taggle. Scott, can you tell us, do you see value and opportunities in linking the CIRSE activities with those of the CTSA? And if you can, give us any thoughts on how we might be able to be more proactive in that, especially from an FDA perspective.
Sure, and thanks for the question, Dan. Yes, yeah, actually, early on, a few CTSA hubs had partnered with the early CIRSEs to kind of propose a collaborative consortium, you know, I guess a pilot, so it wasn't the full networks, but maybe it was overly ambitious. But yeah, I think there's areas to collaborate both on the research and the training side. We've we've done things more ad hoc with different student initiatives, student competition around regulatory science, some joint internships we've set up with some of the between the CTSAs and CERCES, but again, a little more case by case. I think on the research side, there's we've looked at, you know, maybe some there's the collaborative innovation awards that NCATS offers for the CTSAs, maybe partnering with one or two CERCES on a particularly, you know, relevant regulatory science topic. But yes, I think there's, as I hopefully outlined, I think there's a lot of areas of alignment on some research and training needs that we could be more proactive around. And whether that's piloting or seeding a few initiatives might be a good way to start.
Actually, Scott, let me ask you about that, because in one of your slides you mentioned the curriculum around regulatory science, and I wonder what your thoughts are on what some challenges might be that we could help tackle as a part of this collaborative community to rolling out a regulatory science curriculum. I know that you've been actively involved in trying to do this for the CTSAs. How might we think about that as a focus of one of the prongs of our regulatory science collaborative community? What might be some challenges we have to tackle early on?
Sure. I mean, I think part of what we've learned is because different institutions have created, you know, formal masters, some even have PhD programs, some have certificates, some it's more just courses or modules. And so that's part of what's driving us to do the needs assessment. So I think a better sense of, and there's a lot of diverse needs, so we may need to kind of create a modular curriculum that people can utilize for different purposes. But I think for us, having a better sense of what are the current and, you know, evolving needs for regulatory science training, and at what stages, and, you know, because people have needs at different career stages. So I'm hoping that will be informative for us. And so maybe, you know, thoughts you have about that, or good ways to go about doing some of that needs assessment. And even for FDA, you know, you've probably done a lot of work on what are FDA's needs for regulatory science training, and, you know, for incoming reviewers and scientists to, you know, current employees. And so getting a better sense of that might be helpful.
Yeah. Dr. Zhang, are there any questions from the audience there?
Question from.
And I know I'd gone online and answered Khaled, so sorry, I jumped the gun on that one. But
It's okay, we'll come back to that. Dr. Zhang? Or Klaus, are there any?
Anybody know?
I think people may be scared about using the mic with all the feedback.
Okay, that's no problem. Well, then I'll take the opportunity, if that's okay, to ask a question from you two, and thinking about, you know, one of the things I'm thinking about with respect to this collaborative community, and I know this will come up in the next panel, is what are some of the ways we might consider how we optimize or maximize FDA's participation in these different activities? Because what we heard from our success stories this morning, what we heard from both of you in your presentations, and I know in our office, that these regulatory science collaborations are really instrumental when we have FDA's involvement. But FDA is such a resource-strapped organization, we can't be in all places. So what are some ways we might be able to optimize or maximize, do you think, from your perspective, FDA's engagement? Do you have any thoughts on that?
Maybe just make one quick comment, then turn over to Klaus. But yeah, I think it ties into training. One thing we've struggled with is to have case studies, which you highlighted in the previous session. So really a way to kind of capture those succinctly and disseminate them, and for academic institutions to use them as a training resource. I mean, some of us have taken advisory committee meetings from, you know, previous products and kind of package those as a use case to discuss an example of regulatory science. So again, more on the training side, but to utilize these kinds of, you know, great cases we heard about earlier as a training resource is one option too. But.
Yeah. Okay,
Please,
so
Klaus.
the other aspect of what Scott mentioned is not just providing the formal training, because that's part of the equation, that there is a recognition that that's a scholarly line that people can take, that regulatory science is actually a professional thing, not a professional option. That's one. But then on the flip side is making sure that whenever there is a drive to generate any kind of an idea that could solve tangible problems for medical product development, that it goes both ways. So if the agency is approached, that the agency can then, because there's a lot of stuff that is uninformed, not because there's anything wrong with the science, but just because the procedures and the mechanisms are not understood and things of that nature. So the agency can act as an orchestrator to point those groups to places like Scott's shop or even us or other places in academia, where those people can get some orientation as to the who's who and the what's what for these kinds of regulatory interactions. So that when things become formal at the agency, it doesn't become a chore for the agency, and with a big churn of time and money spent on something that is not really that mature from the regulatory science standpoint to advance. I think that both sides of the fence actively contributing to ensure that there's a proper orientation as to the regulatory science components of anything that is intended to solve an unmet need in medical product development is recognized and is properly placed.
Yeah, and just to complement that, I think, as you were saying, Klaus, beyond the formal training, recognizing all the challenges and having people formally do a rotation or an internship or fellowship at FDA and industry, but the more people who have that kind of direct experience, hands-on, beyond the coursework, can really be incredibly valuable. But again, knowing from personal experience, recognizing the challenges to do that even for a short period. And I know FDA has established more fellowships, internships, rotation opportunities, but those are really valuable.
Well, I find it interesting that you both emphasized training as such a critical piece to all of this, and in particular, a formal recognition of training, which I don't believe we have at the agency at this point. And thinking about other ways we might strengthen this regulatory science collaborative community. Scott, one of the things in your presentation you mentioned is this patchwork of collaborations, and I think that was highlighted again from the success stories in the morning and even in Klaus's presentation. How might FDA itself or a new regulatory science collaboration, maybe with like the Reagan-Udall Foundation, for example, you mentioned FNIH, the Reagan-Udall Foundation for FDA serves a similar role for the agency. How might we think about this regulatory science collaborative community serving as the thread to stitch that patchwork together? Now, you mentioned training at the onset. What might be something else we need to tackle to make that happen?
Well, right. I mean, yeah, go ahead, Klaus. Did you have a comment?
Yeah, well, my take on that one is also the aspect of putting some incentives in place. And this would require some tighter connectivity with places like NIH, so that the grant incentives also include for those efforts of let's call them hardcore science that are intended to have potentially a practical application, that the regulatory component is not viewed as an afterthought, but it's front and center for everything that needs to be done as part of those scientific endeavors. And so that's one component of the incentives, because if people see that they will get funded if they become more competitive by having the regulatory strategy front and center, then that's going to spur them into action to seek those opportunities to get informed and trained and collaborate with entities that already have a strength in that field, so that the regulatory components front and center. So that's a big one in my mind. But Scott, you go ahead.
No, I completely agree, and those incentives are critical. And I was just thinking as you were describing that because there's obviously a lot of collaborations at different levels, but I know there was a joint leadership council, but I mean, from individual projects to the FDA commissioner and the NIH director, I know there was a series. I think Dan may have been involved in that. Kind of setting a broad vision and then actually all the way through to supporting particular projects or priority areas. When you have that kind of buy-in across levels, it sends a message and it helps the community focus on particular areas, but tied into what Klaus mentioned as well.
Thank you for saying that. In fact, I think that highlights one of the areas that I know Dr. Dan Tagle has been trying to engage with FDA on with an upcoming request for applications in a particular area focused on microphysiological systems. I know NIH is keenly trying to fund research that has the opportunity to actually cross that finish line, both on the translational science, the bench to bedside piece, but then also passing all of those regulatory hurdles, right, and actually making it to market. And so thinking about how we can support all those steps of the way, I think that's a really good example. Ties in both what you said, Klaus and Scott. So thank you both for your input on that. I didn't see any questions that came in specifically addressed for me, but we will be able to address any questions you might have in the discussion part of the next panel. So at this point, Dr. Zhang, are there any, or Klaus, are there any folks there waiting to ask questions?
Not really.
Okay. All right. So what I would like to propose then is we take a break. We get our panelists ready for our next session that starts at around 10:45, plus or minus a couple minutes, pending any additional technical difficulties. You'll hear from folks at the NIH from 2 different offices, in addition to more details on the Critical Path Institute, which you saw is instrumental working along NIH as well, and of course from the Reagan-Udall Foundation for FDA. So come back in a few moments. Thank you, everyone.
Thanks, Tina.
Thank you.
All right, so welcome to the 3rd session of our Regulatory Science Roundtable. In this session, you are going to hear from different stakeholders who have different collaboration models for engaging with stakeholders in the regulatory science ecosystem. First, you're going to hear from Chris Sasiella from NIH's SEED office. Then you'll hear from Dr. Angeloni from NIH's Blueprint MedTech Group. You'll hear from Klaus Romero on the Critical Path Institute. And then last, you'll hear from Susan Winkler about the Reagan-Udall Foundation for the FDA. We've left plenty of time for Q&A, and this is where we hope to kickstart the discussion on how we can build from the lessons learned to accelerate innovations from translational and regulatory science research. So with that, I'm going to pass it over to Chris.
Excellent. Thank you very much. I am going to try to do just a screen share. Well, hang on one second. Sorry. I hate when this happens. Of course, it only happens when you're live and in person. Let me go to the slideshow mode, and if someone could verbally affirm that I am sharing presenter mode.
Yeah, we're seeing your the opposite.
You're seeing the opposite. Okay, so then
so swap your display settings. Do you know how to do that? Oh.
I believe I do. I'm going to share screen 2, and that should take care of it. Is that better now?
Perfect. Thank you so much.
Excellent. Thank you so much. First off, thank you very much for inviting me to be part of this event today. It's been a very exciting morning so far. I know many of you through the years of working together, and it's been a delight to hear about how each of us is doing our own part in contributing to the evolution of regulatory science. What I'm going to talk about today is this new office in NIH, the Small Business Education and Entrepreneurial Development Office. We call ourselves SEED, and how we have contributed in our own way to the evolution of regulatory science and put it into action. So SEED is, as I mentioned, the Small Business Education and Entrepreneurial Development Office. We have quite a few activities that we are engaged in. We do manage the HHS Small Business Program. That is the foundation of the office, is what we grew out of. But we provide a lot of education, not just about the small business program and all of its attributes, but also information that's really important to innovators in the life science space. Webinars, conferences, on our website, through our help desk. We have subject matter experts who provide consultations to NIH-funded innovators to help them understand those critical steps to help them move their concept into clinical and then adopted practice. We do that for both small business awardees as well as for academic and other translational program awardees. And we have a rather extensive entrepreneurial development focus through not just those educational opportunities, but also through coordinating a number of proof of concept networks, one of which I will highlight later on in my talk. But when you think proof of concept network within NIH funding realms, you can think of things such as the CTSA program that Scott mentioned earlier, or some of the major networks that come out of the National Centers for Accelerated Innovation or the Research and Evaluation and Commercialization Hubs, the Blueprint Network, the epilepsy or the asthma networks. There's a whole lot of coordination that NIH does that really does a lot of proof of concept testing, from lead discovery all the way through clinical proof of concept. And we also do our best to enable investor and partner relationships so that when that NIH funding runs out, as we know it will, at some point the NIH will continue to be funded, but any given project is going to end up needing more funding than NIH can provide. Within that space, then we do our best to make those connections with the strongest of the innovators in the NIH portfolio to those investors, be they angels, VCs, philanthropic funds, strategic partners. We help facilitate that. Now, all of that sounds great, and people get really excited when I'm in person at a meeting to come talk to me and learn more about this, but really, it's not me that's the power behind this. It is the innovator support team itself. We have 6 entrepreneurs in residence. These are seasoned life science professionals who really, truly understand. They have worked in large pharma, Fortune 500 companies, Fortune 100 companies. They've started their own businesses. They've had successes and failures. They know what it takes. They have the connections to help the NIH innovators really move their technology along at that early stage where many of our innovators live, and at that more advanced stage where some of our innovators live. We do have a subject matter expert available to discuss some of the intellectual property challenges that come up. Now, many times our innovators are funded by or are primarily associated with academic institutions. So they have their tech transfer offices and they're well taken care of from that perspective. But for those small businesses or independent innovators that come in and receive NIH funding, it's useful to have someone to sort of bounce those ideas off of. And then, of course, we have regulatory support, and we have payment experts also on staff. And these people, like myself, may have spent some time at FDA, either through fellowship programs or as career paths, and have also worked at CMS or been aligned with CMS and other healthcare groups, payer spaces. So really, we try and bring everything but the science comes in to the innovator support team. Now, what I'm going to talk about for the next few minutes is how all of this expertise really converge to help a specific emergent need. Earlier today, we heard Tina talk about the new initiatives of the regulatory science priorities, and the public health preparedness and response was part of that, right? We talked about, you heard Leonardo and Scott talk about how there's an emphasis on team science and specific examples of developing resources. Klaus, similarly with the biomarker qualification, came in and talked about how these tools that enable development can be collaboratively developed. Well, one of the reasons this is a hybrid meeting today is because of SARS-CoV-2 and the public health emergency that has been in effect for some time. NIH was tapped on the shoulder at the very beginning of this pandemic and asked to create a whole system of funding mechanisms to rapidly accelerate the development of diagnostics and therapeutics and vaccines to address this emergent public health need. And we leveraged one of the existing proof of concept networks called the Point of Care Technologies Research Network. This is funded through the National Institute on Biomedical Imaging and Bioengineering, but expanded to encompass so much more than that during the past 2 years. The PopTurn network, as I'm going to refer to it from now on, just to save myself some breath, was established in 2007. It expanded tremendously during the pandemic. And really, what we were able to leverage so effectively was the validation core, the clinical studies core, and then the deployment center to help with supply chain challenges. Now, you're thinking to yourself, that sounds great, Chris. What does that have to do with regulatory science? I'm telling you, this slide represents regulatory science in action, because the innovator support team members that I showed you on the prior slide, the network here, plus FDA, plus CMS, plus BARDA and DARPA, all coordinated to focus on the development and the acceleration of these technologies. Understanding what are the actual biomarkers, what are those qualifications, what are the limits of clinical detection and the size of the samples that would be needed. Members of the regulatory and reimbursement team on the innovator support team, those subject matter experts, participated in weekly calls with Tim Stenzel's group at FDA. He's the leader of the Office of In Vitro Diagnostics and Research that was leading the development of these diagnostic technologies. We worked with FDA, literally our team members worked with FDA, provided feedback on draft templates, helped them understand what innovators were seeing, because those EIRs that I just showed you were working side by side with dozens of investigators through the RADx who were funded through the RADx program, and really help them take the innovator questions to FDA and help bridge that communication gap that can sometimes happen. So what did it take to make all of that happen? Obviously, there was a very common mission focused on addressing an emerging global health threat, right? There was a very strong stakeholder buy-in from FDA's CDRH, Office of In Vitro Diagnostics. And there was that collaboration with the RADx steering committee that included investigators, awardees, subject matter experts, right? We worked together with these people to understand the prototype validation requirements, the clinical core validations, what would be a valid sample even for doing that validation. We helped define those standards. So we've heard that already today, that the development of consensus standards, tools, biomarkers, and publication of guidance are hallmarks of regulatory science. And this really is sort of the cutting edge of where my office, the SEED office, and my team, the innovator support team, are going. And these contributions over the past 2 years have just completely revolutionized, I think, the way that we think about collaboration and developing public-private partnerships. This is a super exciting time for me to be part of a government office that has this capacity. And I very much look forward to hearing the stories and sharings of my fellow panelists, and to entertaining your questions in just a few moments. In the meantime, I know these decks are going to be shared at some point in time. I did have a little trouble uploading mine, so I apologize for that. But if you are interested in finding out more about the resources of SEED or hearing about our upcoming initiatives, please check out our website. Right now, it may not look like much, but I can tell you I have a ton of educational resources, knowledge guides for innovators, case studies that we're going to be publishing to help the innovator community understand the regulatory processes and reimbursement processes and business processes of taking the concept and bringing it to public benefit. Thanks so much.
That's terrific. Thank you so much, Chris, for that excellent overview. Leonardo, you are up if you're still there with us.
I'm here.
And for the folks online and in the audience, I encourage you to drop your questions for us in the Q&A box. Thank you.
Can you see the screen, Pro?
Yes, thank you.
Okay. Good morning, and thank you for the opportunity to provide an overview of a new program that was established in collaboration with many other program officers throughout the NIH. The name of the program is the Blueprint MedTech. I will be providing a short overview. We have recorded webinars, full slide presentation in our webpage, and certainly available for any further questions and follow-up. The program was launched last year, and as you can see, is a collaboration of is part of the NIH Blueprint initiative, which involve over 10 institutes and centers that are signed up on the program, with the leadership of National Institute on Drug Abuse, National Institute of Biomedical Engineering, National Institute of Neurological Disease and Stroke, and National Institute of Mental Health. The program is conceived as an incubator that aims really to accelerate the development of cutting-edge medical devices intended to diagnose and treat disorders of the nervous system. The idea and the mission is really to catalyze the translation of novel neurotechnologies from early stage development to first-in-human clinical trials, and to accelerate patient access to safe and effective and groundbreaking medical devices. And the program, if we look at the overall product development path for investigators, the investigators have completed their proof of concept stage. They have completed basic research on mechanism of actions, and they are moving toward the translational path. I offer this program to provide technology optimization, and then once a specific disease or disorder is identified, move forward in the translational part with the studies, which include preclinical testing and early clinical first-in-human studies. That's where the program comes in. So, and then later on, through other type of support, of course, continue along the path of the product development with further pivotal studies for human subjects. This slide is quite complex, and this is what we have available again also on our webpage, but provide the structure of the program. And the idea is not to provide only non-dilutive funding, but of course a critical component of the program, just like for many other funding opportunities, but also free to the investigators additional resources that are provided by NIH through different mechanisms that we believe are fundamental to have successful development of novel medical devices. And the program is composed of, there are 2 stages and 2 entry points, if you will, for investigators. Early stage seedlings, this is essentially managed by centers or hubs. The solicitation was published last year and is currently being through the administrative review process. And these hubs would essentially solicit early ideas still on the technology development phase. But if there is an investigator or a company that already have passed that phase, then there are specific mechanisms Some they are specifically dedicated to small business, others they are open to all investigators. And these are phase mechanisms with very focused milestone-driven approach that would allow funding. And as I mentioned, additional resources in terms of business development, regulatory support, biocompatibility or animal studies, and clinical support. As you can see, the idea is really along what we have been discussing for the entire morning, which is really the collaboration between investigators, NIH, FDA, and industry, and in pushing forward devices that are new. We don't limit, but the idea is we expect devices that would be possibly going through CDRH regulatory path as PMA or de novo. Some 510K maybe also be acceptable depending if there are new specific regulatory questions. The idea really is to support novel devices and then open the field to then others that would take advantage of a new path that is combined. As I mentioned, we have our webpage where you can find information about funding opportunities and additional resources. This slide deck is in a full webinar, and as well as other webinars that provide additional details. And of course, you can contact us on our email address. I'm happy and look forward for the discussion today as well. Thank you.
Terrific. Thank you, Leonardo. That overview was very complimentary to Chris Saciella's presentation. Next, we'll hear again from Klaus. I hope he's able to join in the room. Instead of focusing on a case study, which was awesome, for that, he'll give us an overview of the Critical Path Institute itself.
Thank you. And thanks to the AV guys here in the room, they've been scrambling. So thanks for that. Can you see the slides and can you hear me, Tina, on Zoom?
I can hear you well, which is terrific, but we cannot see your slides yet.
Okay, they're working on it. Here we go.
Okay, there they are. And for those participants, oh, perfect. You even made it full screen. Excellent. Thank you very much.
Awesome. Okay, so after the presentation during the break, I got a lot of the questions that I'll address during this presentation. So again, what is a Critical Path Institute? We were, and it's in the name, we were started in 2005 in direct response to the Critical Path Initiative from the FDA. The CPI is the abbreviation of the Critical Path Initiative from the FDA. Our acronym is CPath. We're the Critical Path Institute, and we were started specifically to help realize the vision of the Critical Path Initiative document from 2004. I know that that document has been updated, but even the original form from all those years ago is a really visionary document, and it has withstood the test of time, and it is really a charter for how to transform medical product development. And so what is it that we do? We generate this neutral environment. How do we generate this neutral environment? We have the legal infrastructure, we have the scientific infrastructure, and the regulatory infrastructure to create this neutral environment where companies, regulators, the patient groups, and academia, but other government agencies as well, can come to the table and share information that they would not be able to share otherwise. For what purpose? To transform that information, those data, into actionable solutions that can help address specific bottlenecks in the drug development process. And that's the key component of that, collaboration and partnership. And so through that neutral environment that we have provided, we've been able to provide a home for collaborations across many organizations that have a piece to contribute to the overall puzzle of really solving specific bottlenecks in the process. Now, those collaborations take tangible forms in the form of mostly consortia. Not all the things that you see on the slides are called consortia, but they are still collaborations or public-private partnerships that were established to meet specific deliverables for unmet needs across therapeutic areas or just types of solutions. So what we have are collaborations that are either disease-centric, but solution agnostic. The PKD example that I showed is one where we're talking about data, modeling, biomarkers, et cetera. So all those are solutions, but it's all around PKD. There are other efforts that are solution specific, but indication agnostic, like the PRO consortium. All they do is develop novel transformative patient-reported outcome instruments for a number of indications, but the core of that collaboration is around the type of solution that is the PRO instruments. And then we have collaborations that are both solution agnostic and indication agnostic, like RDC-ADAP, which is the data platform for everything rare disease and orphan disease data. But we really need the data, and this is great news, because as I was saying, now we're past the 500,000 individuals that are in data that have been contributed to us from clinical trials from industry, and that includes not just the control arms, but the active treatment arms as well, observational studies, patient registries, but now importantly, electronic health records. More than 300,000 babies represented in electronic health records from the neonatal intensive care units that are going to be leveraged next to generate a couple of transformative solutions for neonatal drug development. But if you look at it, you might say, well, you guys are trying to boil the ocean. Well, it looks like that on the surface, yes, but it's not like we wanted to tackle on all those unmet needs. People are coming to us. So if there's anybody to blame, is the stakeholders that come to us, but it's a great problem to have, and we never turn them down. We just listen to them very intently. But in reality, if you peel the onion, we're really talking about 5 core competencies that we have, which are the hexagons that you see at the top of the slides. Everything that pertains about data, biomarkers, modeling and analytics, clinical outcome assessments, and that of course includes PROs. But importantly, the wrapper for everything, regulatory science. And we just happen to have concentration areas, conduit areas where the core competencies flourish. Every single solution that we put out for any therapeutic area, for any drug development or medical product development area, is an expression of one or multiple core competencies that we have, and we don't deviate from those. The other question that I got is, how do you fund all that? Well, here's the answer. So each one of the consortia that you saw in the previous slides has its own funding mechanism. The most prevalent funding mechanism is actually example number 3, which is a combination of hybrid funding from one or multiple grants, but industry fees or member fees that every member of each of the consortia pays to be part of an individual consortium. There are efforts that are funded exclusively by member fees, and there are a couple of examples where the funding for the entire effort comes from a single source. Big credit to the FDA. The Rare Disease Cures Accelerator Data and Analytics Platform is currently solely funded by the agency. So just big sign of recognition to the agency for that. But what is at the core here? The flexibility. The flexibility to do what? To understand exactly what the unmet needs are from the community. And the community is industry, the patients, the regulators, the academics. We spend a lot of time listening. I'm a clinician by training, and to me, it sounds like we need to listen to what's happening to the patient being the ecosystem in that particular indication. The narrative of the patient is going to be the narrative of the patient. That doesn't mean that that's the diagnosis. If somebody comes to you and says, oh, I can't sleep and I have headaches, you can't just automatically say, oh, it's easy, insomnia and migraine, so I'm going to prescribe sleeping pills and pain medication. Wrong. That's not the way to do it, because just saying I can't sleep and I have headaches are manifestations, signs and symptoms of many potential underlying problems. And you need to understand what the underlying problem is before you start formulating a treatment plan. That's what we do with the different areas. All to do what? To generate those solutions, but those solutions need to be adopted. And I have an industry past. Not that I'm ashamed of it, but I do have an industry past. And when I was in industry, I was reading the literature, keeping up with the science, and I saw all the new methods that were being proposed. And I thought, well, for our next trial, that's the kind of methodology that we need to adopt. And I printed the papers, I built the narrative, I went to upper management, and 99.9% of the time, the answer was no. Are you crazy? That science that you seem so enamored with can introduce another 6 months or 12 months worth of review with the FDA. We don't know what the FDA thinks, and I'm making air quotes for those on the phone. We don't know what the FDA thinks about the science behind that solution that you like so much. Well, hence the process that FDA built and EMA built to review these solutions once. That's it. And at the end of the day, what do you get? A letter from the regulators saying, we have reviewed the science behind the solution for this unmet need. And yes, it does solve the unmet need. And we encourage people to use it. Had I had that in my industry days, I would have just have printed one letter, and I would have gone to upper management and said, the FDA is encouraging us to adopt this new solution. So the end, let's adopt it and let's make things move forward more efficiently. So examples of impact. Tuberculosis. The solutions that we've generated resulted and contributed to the first approval of the first new drug in 50 years, and then 4 years later, the first regimen for tuberculosis. Alzheimer's disease. A whole host of quantitative solutions that are transforming the way people are thinking about clinical trials in Alzheimer's disease. PKD, the example that we just went through before. In biomarkers, a collection of composite biomarkers that are really transforming how you think about dose selection during drug development in terms of kidney safety. Parkinson's disease, a series of quantitative solutions, but importantly, how to think about imaging markers and how they can be used to optimize the selection of patients for these trials so that those trials are more efficient. Type 1 diabetes. Again, biomarkers that can help really select patients for prevention studies. Prevention of type 1. This is not type 2, and this is not insulin treatment. This is prevention of type 1 understood as an autoimmune disorder, and you need to understand that you need to get people into the trials that don't have a diagnosis. That's not an easy proposition because you need to recognize who is really at risk to then do a prevention trial on. And in terms of patient-reported outcome instruments, 3 areas where the voice of the patient has really transformed how people are thinking about the trials that are being done in each one of these diseases. And I'll conclude with that. And hopefully I see you shaking your head in the room. I hope that clarified all the questions you had, which were great questions. I'll stop with that. Thank you.
It was an excellent overview, Klaus. Thank you very much. I have several questions for you, and I look forward to asking them during our panel. And our last presenter for the morning, before we kickstart our discussion, is Susan Winkler from Reagan-Udall Foundation for FDA. Susan, are you there with us? Oh, she's right on top of it. Great.
I am, and I think you can see what I'd like you to see on my screen. So thanks so much, Tina and Guijan, for the invitation to join you today. I'm excited to join this panel and talk a bit about what we've been doing at the Reagan-Udall Foundation for the FDA. And I'll note, I want to confirm you can see the slides in the room. There'd been mention of the foundation earlier, and just to confirm, we're a nonprofit, non-government organization that Congress created in 2007 to help advance the mission of the FDA. So we don't undertake any activity that doesn't relate to something that would advance the mission, and a core component of that is often in advancing regulatory science. So I'm going to talk about a community that we've developed during COVID-19 to advance regulatory science, particularly as it relates to real world evidence, and just use that as an example of opportunities for collaboration as facilitated by the foundation. So if I'm going to talk about the COVID-19 Evidence Accelerator. And the Evidence Accelerator was this gathering, because it's still continuing, a gathering of the real world data community, engaging folks from FDA, from other government agencies, and from the private sector, particularly data and analytic partners, so folks who hold real world data from electronic health records, from claims, from patient devices that generate information from patients and other repositories, and was intended to say, how do we try and keep pace with the real world data that the world was swimming in to better understand what was happening with FDA regulated products as they were being repurposed, as they were being created and made available under emergency use authorization, what was happening in the real world with the use and the performance of those products. So you might assume, but to underscore, there was an intent here, a real world need to say, we've got to keep pace with what it is that's happening, and to use real world data to advance and expand limited clinical trial resources, to keep track of what was happening with these newly authorized products. And then the idea of the community was to have that rapid shared learning so that we could better understand what was happening. There were questions, if you looked at electronic health records and were trying to assess the severity of the patient, what level of oxygen was supplied to the patient. That's quite inconsistent in many electronic health records, and so there was work through that. Or things like, if a health record does not say that I've received a COVID-19 vaccine, does that mean that I have not received a COVID-19 vaccine, or does that mean simply that the data about that vaccine isn't included because I may have gotten it in a facility that doesn't typically report to an electronic health record? So a lot of discussion about some of the nitty-gritty of the data, and then higher level issues as well. So in this community, we built a community that focused on therapeutics and vaccines, as well as diagnostics, and then some cross-cutting discussions related to oncology and some work in post-COVID, and pursued two different efforts. One, to be able to have those collaborative conversations, and a second, to pursue specific research questions and then present those results in parallel. And what we found with both is that they were extensive learning communities where the private sector would share, here's what we're seeing and the challenges we're seeing. Public sector would similarly share, here's what we're seeing and the challenges that we need to navigate, and work through some of those issues together. So I'll just note here one of some notes that we developed about what were some of the things that helped. What did we do in accelerating evidence? We found that the COVID-19 Evidence Accelerator community spawned other collaborative efforts. So there were offshoots that worked through things. We found that there was this opportunity to trust and share, to do work together, to identify obstacles and overcome those, to collaborate and share information rapidly, and then to learn about the different work that was being pursued. So we continue to gather now on a monthly basis to say, what is it that's happening within the pandemic that we can pursue, and to continue to explore what it is that we might see it moving forward. So I'll share that, and I think, Tina, it might work well as we move to the question section to say, where might we need a routine engagement to explore emerging concepts, a safe space for public and private center interaction, this opportunity to build familiarity with new concepts and test assumptions. This is what we found we were able to create in the COVID-19 Evidence Accelerator, and may have applicability as we're looking here to say, how do we continue to advance regulatory science and bringing some of these opportunities together? So Tina, I attempted to go quickly so we could get to the Q&A, because I know that's the most important part of our discussion today. So with that, I will toss it back to you.
Excellent. Thank you so much, Susan. If we can have Chris and Leonardo and Klaus and Dr. Zhang join us on the screen. And I hope Steve could facilitate that in the room. Thanks to our speakers. I really appreciate you guys jumping right in and staying focused in the interest of time and maximizing our discussion. Okay, I see us all on the big screen. You guys can pin the panelists if that's helpful for you on your screen. So I know that, let's see, Scott, you did a great job of kickstarting some questions for folks in the chat. Why don't, Scott, we start with the question you have for Klaus, because it's certainly one that I have as well. How can we more broadly disseminate the results from the consortia and programs into the community? I think that we heard about these success stories today. How might we put those success stories out? One of the comments Scott made was maybe it's a formalized training program. So how do we memorialize that? How do we make those results available to the community? And if you are doing this, if one of your organizations is doing this already, how are you doing that, and how can we build on top of that?
Admission.
Susan, why don't we start with you, and then Klaus, it's going to take you a second to get on.
I'm on. Can you hear me?
Oh, excellent.
Excellent. All right. So yeah, thanks for that question, and thanks for that push. I need to make a full admission that I've had to make several times, and it's an admission, but it's at the same time a recognition that we have not done great at disseminating our impact and our stories. Our branding and our marketing have fallen way back to the back seat in favor of the collaborations on the science. So that's recognized. How do we disseminate that? We're open to anything and everything. We're working right now with a scientific dissemination and literacy firm to try to solve that also from our end, but in the spirit of collaborations. If there's any avenues that you guys in the panel can shoot our way, or a mechanism that that we can leverage, we're all ears, because we do recognize that in the interest of just making sure that we do the work, the dissemination has suffered. So there you have it.
Klaus, I'll say from FDA's side, you are not alone. I know that is one of the challenges we face in our office. So maybe this is really something we all come together on, and we get ourselves over to the National Academy of Sciences and say, hey, we need your help to lead like disseminating all of the good science, because we're all strapped for resources and it's so hard to prioritize that. Susan, jump in, please.
So I was going to jump in to say that one of the things we've seen with the Evidence Accelerator, and I think it has some applicability here too, is that we often have that tendency to say, when are we at publication stage? And what we explicitly adopted in the Evidence Accelerator piece was to have closed door meetings, so you keep things within the community, but then after each meeting, there's a public posting of a summary of the discussion. So you're sharing the output and seeding the community and saying, here's what we're talking about, and trying to generate that discussion as you go along. Because we did that routinely, then folks became more comfortable saying, okay, they became more comfortable to talk about things earlier in a process, as well as to do some of that public sharing. It's by no means a cure-all, but it was very helpful. A second piece is that we invested a fair amount in visualizations. So trying to pull together something that would emerge from a couple of different meetings and say, how can we turn that into a story? And that visualization we found has been incredibly powerful. So just a couple of thoughts that help. I think they're baby steps towards what you're looking for.
Well, that's really helpful. Chris, do you want to add anything, or Leonardo, before we move on to our next question?
I agree dissemination has to occur along the way. And then, of course, as mentioned before, it takes years for guidance. It takes years for even a publication or work that has been done. I do see a question kind of related on the chat, which is about who owns where the project, if there is a project, let's say developed by Blueprint MedTech, supported by Blueprint MedTech, how would you know about it? Information is on the NIH, but you need to wait for that project to end. But what we did in other cases, including case studies earlier today, is sharing even through abstract, through conferences, the stories, or why communication after a meeting along the way, and then building the community so that by the time the guidance was published 5 years later, the community was already aware of all the work.
And I definitely agree with that. I think one of the challenges we have is that we often only talk to those people that we know, right? It can be very hard to reach a new community and to raise awareness about the emerging trends in any field, and certainly regulatory science in particular. I think one of the things that we could take action on immediately to help expand that reach would be to make a commitment to follow and scrape and repeat information on social media from as many relevant groups as we can. I know the SEED office has a LinkedIn profile and a Twitter account. I don't manage them, but I certainly do my best to promote awareness of the things that come through me. I send them off to our social media team and they propagate. And I do suggest places for them to follow, scrape, and tweet on a regular basis. I think if each of us within our own realms, whether we're with industry, academia, a foundation, whatever, were to really take that same approach, you never know what influencer is in your circle of followers that might really help raise the awareness and have that one-off conversation at a cocktail party if we ever go back to having those.
Yeah, I think there's something important to what you're saying, Chris, and that, you know, each of us has a stake in making sure we're communicating to our networks the important work that's happening in this domain. And certainly our office is really keen on thinking about ways to capitalize on that. But I do think this idea of dissemination, that work is really, really critical so that people can learn from our opportunities and our successes. And also places where we're not successful. That's a whole nother bucket I don't really want to open up here, but I know you all know that learning from failures is just as important as learning from successes. There was one concept. Susan, did you want to jump in?
I was just going to note that I think learning from failures is easier when you've been having the tough conversations along the way. So if you've got, you know, if you're having the conversations and sharing the information, then it becomes easier to say, oh, here's what we thought we saw, and here's what was actually there. So that comfort level, and then if we could share more about not only what's great, but also what didn't work.
I want to also just, before I move on to the next question, Dr. Zhang is also in the audience. And Klaus, I don't have any visual to be able to see if you're trying to jump in as I can with the panelists on Zoom. So you'll have to just interrupt me if you want to jump in and say something if I can't see you. So don't be discouraged. You either, Dr. Zhang. So with that, there's a
We're
really
standing by.
important,
Can you guys hear us?
Yeah,
Okay.
Great. Nice to hear you. Wonderful.
I want to, oh, here, we actually have a question in the audience. You want to ask or I translate or can relay? Can you guys hear him?
I said, my name is Raj Kazineth, and I'm from Johnson & Johnson. And my question to the panel, maybe specifically to Klaus, with regards to the Critical Path Institute and some of the information that was shared, as well as the NIH Blueprint program, perhaps. Maybe that's the focus of my question. With regards to disease states that, again, Klaus described this, there are situations where you know there's a patient value at the end of the intervention, but companies just can't get into the space because the numbers are just too large in certain disease states from a clinical translation perspective. If I want to be specific, it could be something like say, an infection rate for a certain procedure, right? So if you're at a prevalence at about 1%, then the intervention, to take it down to maybe 0.5%, needs to be proven out clinically at probably something like 10,000 patients, right? To show that type of difference. Under those kinds of scenarios, even though the percentages might be low, like the 1%, that could translate sometimes to 100,000 patients, right, annually. So under those kinds of scenarios, are there thoughts about how we would go about building an ecosystem to address that type of clinical translation, such that the companies who want to bring such innovation out there are able to do it in a way that's sustainable, right? Because currently there might be some things, the technology I think is out there, but it's how do you get it out there so that there is equity amongst the different stakeholders to be able to make that move. Any thoughts on that, or how these different initiatives have maybe or can enable that?
Yeah, no, that's a great question. I'll start and I'll pass it on to others on the panel. So, and that's a question that we get quite a bit. We have that challenge. We need to run the trials, and we're not saying that we're asking anyone to lower the bar of evidential requirements, but there is a challenge with identifying the patients. What kind of metrics are we going to run? How long are we going to try to recruit for individuals, et cetera? And that's where your term ecosystem comes into play, right? Because that's when you need to understand what happens in the trenches, in the clinical environment where those patients might be identified. If it's a disease that just happens to have the advantage of having one or multiple patient groups behind that, that's another component of something that needs to be brought to the table. And then the other piece is the notion of how you run still solid evidence generation, but through not necessarily the same traditional approaches. And there's one example to your point that I can cite, and it's actually in the medical device field, and Tina, that's where your expertise truly lies in depth, where, and I'm not going to mention names, but where CRH had the vision to work with sponsors to set up patient registries once a device got approved, so that everybody that got the device was put into the registry. That helped with three things. Number one, the original approval of the registry, the label for that could be expanded based on only the real world evidence gathered from those registries once the device was out in the public and was starting to be used off label. Still gathering that information was instrumental in that labeling expansion conversation. Number two, it provided the patients with the knowledge that that was actually happening, and they started to be vocal and positive and active in contributing their data and making sure that others knew that that was an opportunity for them to contribute information that could be maximized. Power number two. And then third, that also allowed for the assessment of feasibility for new devices to be put into a research environment to start generating some evidence that could be translated as proof of concept. And so that's and who does that? Well, it needs to be done by the ecosystem. Does it help if you have somebody orchestrating that? But then the question is yes. The answer is yes, but then the question is who does that? And that's what I think needs to be grappled with in fora like this one. I'll stop with that.
Yeah, I'd like to add something and kind of pull us just out specifically from say any type of trial per se, Klaus, and kind of build on something you actually said in your presentation, which I found really interesting in thinking about how does a consortium or how does an organization working in this space develop a rubric to determine what we should focus on? Like, how do we make those decisions about what's a priority? And you mentioned that there were consortiums that were solution agnostic, disease centric, or disease agnostic and solution centric? How do we, in this thinking about developing a collaborative community, create a rubric where we can, first gather what are those needs and then decide how do we pick and choose, particularly with emerging topics, and thinking about how do we bring something like that to the table to either engage with FDA or get FDA's participation. I'd love to hear any of you guys, your thoughts on that.
I'll let others go first, and then, because I do have some thoughts, but I don't want to monopolize the mic.
Yeah, Susan, do you want to jump in?
Happy to. So it's, I think that's part of your kind of vision in saying what are the shared values and where do we want to pursue. I think it's got to be part of that threshold conversation, and recognizing there are probably conversations. I would sense from the FDA's perspective that you need to be at a stage where things are becoming real. That it's some of the very early conceptual discussion can be helpful, but where the agency really benefits from the conversation is as things are moving and they can become more familiar with them. So I guess I'd say, Tina, part of it is that conversation of helping an inherently risk-averse agency where you sit to better understand what they, what is happening in an environment, and where there's either a dearth of opportunity, and so you need to have the conversation to stimulate work there, or there's a lot going on and the community is looking for some clarity. So there is an art, I think, in having the right time, but you don't want it to be too late when then an inherently risk-averse agency doesn't have an opportunity to see things develop and help contribute to that development.
And if I can jump in here and add on to Susan's comments, yeah, I've been at NIH now for 10 years, and through that entire time, I've been focused on the regulatory education of our innovator community and our program officers. And I have had many, many conversations over the years about when can I get FDA input on this wacky, crazy idea that isn't necessarily specific product development work, right? And it may come up from a program officer looking for a orthogonal approach to disease management, right? Versus what's currently standard of care, or from an innovator who really believes they're developing a platform tool that can enable the development of multiple technologies. And one thing that I found useful is, and one of the things NIH does really well is we convene, right? We bring people together to have those formative discussions. And so there have been a number of workshops where FDA has been invited as a listening member of the event, not as a panelist, a presenter, someone who's expected to bestow the blessings of FDA on everyone's approach, but really to sit in the room and listen. Sometimes there's a little bit of resistance to that because, of course, anyone who sees an FDA speaker at an event or an FDA attendee at an event is going to come and go, hey, I want to talk to you about my widget. But if we can really sort of frame that conversation properly, it helps FDA start thinking about these things in a timely manner. Because NIH is funding stuff today in academic labs that may be products in 20 years, right? Or maybe prototypes in 20 years. But another really useful tool also, I think, and I haven't recommended this, as I said, been with NIH for a decade, I've recommended this about 4 times, is the critical path innovation meeting, right? So that is a really critical place where you can bring in all the stakeholders. Whoever requests the meeting controls the room, right? You can invite whoever you want into that room, and that is a fully pre-competitive space. It is unique and separate from the pre-sub process, the pre-IND, the interact meetings, which are focused on one very specific product, right? The CPIM meeting is really meant to be that pre-competitive space where you're talking about what would be an appropriate level of biomarker qualification to address end-stage renal disease without having a specific product that you're looking to submit, right? Or a specific trial you're trying to support. So I think there are some really great tools available. And for those people who are funded by NIH and you're kind of running up against this, you might talk with your program officer about whether or not there is a broader interest within their portfolio or across multiple institutes like the Blueprint MedTech program that Leonardo described that really could be fitting that sort of niche space.
And Tina, if I wanted to also build upon that, I think it's key also to, of course, build upon the FDA regulatory science teams and tools, because again, we have done that. We've done that for years. The community, and when it comes to right time, I would argue it's never going to be early enough, because the reality of it is that a community, especially community of small business, is so concerned, so scared to approach the FDA that most of the time they arrive late. And so late to that conversation. It's extremely difficult as a program officer to invite and to encourage small business. They're not in the early phase, they're past the proof of concept, they're really already working in even starting the conversation with the FDA. And as you can imagine, they're already late. We have done that. We have built, when I was with the FDA also, we have built community, and we were talking with them as early as NIH-funded basic studies or mentored developers of program, because the mentality, when you develop from the mentality of doing something that is not just a cool tool, but is something that needs to get to patients. So you need to design with patients in mind, with the disease, what is the need that you are trying to address. And one example that I can bring in building the community and the ecosystem, since I joined NIDA, I've been working, for example, a lot in building a community around extended reality and virtual reality. We have worked with the FDA, we had a workshop. Now we see more companies building this and approaching. There was the first de novo approved by the FDA last year, virtual reality for pain, other indication. But it takes really all the community to start understanding that it's not going to be the perfect device clear right away. It's a work in progress. And the reality of it is that the knowledge, the benefit risk assessment that the FDA does and what they do in 2010 is going to be different than what they're going to do in 2020. That is what happened with the MR. But on the other hand, let's start patients to start to see the devices, provide their feedback, see how they use it in reality. If you think companies that develop, again, the example of virtual reality, they have changed their approach 5 or 6 times within 3 or 4 years. And this is especially true for fast changing devices. This is where, for example, in the Blueprint MedTech we are intervening. It's really, let's build how the technology, let's get the first clear, and then, yes, there will be update. And another pushback that I hear all the time from the community, especially in the field of digital health, digital therapeutics, so diagnostic, this idea that once approved by the FDA, the device can no longer change, so they rather not be regulated and trying to have a consumer product. Again, this is a misconception, because it's like, imagine a pacemaker approved, the software is going to change. And so again, encouraging the community to have this interaction. I think regulatory science offices within FDA, CDER, CDRH, and other are key that allow that early conversation, scientist to scientist, and of course, with the input of the regulatory, but there is a lot of science that can be discussed.
But bringing it back to, Tina, your point in the beginning about the rubric, right? So how do we understand how to prioritize things and understand the feasibility of proposing things? What we have learned over the 15 or 17 years of existence is that you really need to start with hearing the stakeholders out. So you need to start hearing the industry perspective, the patient perspective, of course, the regulatory perspective, but also the academic and the clinical practice in the trenches perspective. There is also this misconception that most of the time those perspectives are not aligned. I would disagree. If you just look at them face value on the surface, yes, you might reach the erroneous conclusion that they're thinking things differently, or that they're not aligned in their thinking, or that they think that the priorities are different. That's on the surface. Once you start peeling the layers and you understand, okay, so what you say that this is a pain point for you, why? Let's dig deeper into that. And a lot of times you reach the same conclusion, that even though this is being expressed differently from a regulatory perspective, an industry perspective, or patient's perspective, it ends up being either very similar or essentially the same thing. And that's when you can take that and articulate that perspective into a properly worded unmet need using regulatory science terminology. And then you say, okay, now I understand what the diagnosis of the problem is. Let's formulate the solution. What kind of solution would work to solve this? And that's what gets you into the, okay, what kind of data do I need? Do those data exist today? Or do we need to go generate the data? And how long does that take? And how much is that going to cost? And if the answer is yes, for this solution, it'll take us 15 years and $1 billion, versus for this other solution, it might take us 5 years for 5 million, then that really paints a clear picture for everybody. And that's, again, back to the notion of, do you focus on something that is more solution-centric? Do you focus on something that is disease-centric but solution-agnostic? And that's how we've learned to do this in that collaborative fashion. But back to the previous point about us being, and I hate that, the best kept secret of certain collaborations. How do we bring that to the fore and share those learnings with everybody, so that, for example, if they're planning a CPIM meeting, a clinical path innovation meeting, let's understand how to best make sure that you have a well thought out set of cool ideas to present to the FDA in a fashion that makes the CPIM, extracts the maximum value of the CPIM interaction, as an example. Or there's people up in arms with signs in front of Congress saying you need to change this. Okay, let's take them in and understand exactly what the unmet need is, because it's not like the government is the enemy necessarily all the times. And then you can get more consensus as to how the solutions could be driven. And I'll stop with that.
Yeah, and I think one of the things that I heard from Leonardo, I'm using this opportunity, this panel meeting, to think about places where we have infrastructure in place from all these different collaboration models, and how not just to think about the gaps in regulatory science, but to think about the gaps for meeting the needs of the regulatory science with respect to these different collaboration models. So finding the opportunities to overlap and collaborate. Leonardo, you said something about, one of the reasons that in the success story you mentioned, there was a regulatory science team at FDA already thinking about that, right? So right at the top when I'm thinking, well, what are some regulatory science teams that we have in place that are cross-cutting, and we have our FDA scientific working groups. So one of the things that I'm thinking about is, how do we take those working groups that have representatives from all the centers at FDA working on a critical science topic. Each one of those working groups is one of our focus areas of regulatory science. How might we start with a group that's already established, that has research and regulatory people who are on the front lines of this, connecting with our stakeholders outside to hear their pain points, to hear their regulatory science gaps, so that we bring not just FDA's pain points to the table and our regulatory science needs, but we think about how we can build a forum for listening to those from the outside, so that when the agency is putting out its regulatory science priorities, is there an opportunity for us to make sure we have balance, both from what's important to FDA, but also for our stakeholders, because what's important to our stakeholders should also be important to FDA, I think. I see heads nodding, which is really great. Any thoughts about that as we're closing out our Q&A session this morning?
I couldn't agree more.
Yeah, same. I agree entirely. I think one of the big challenges with that is that as soon as FDA raises its head and says we want input on how to stimulate regulatory science in this space, then everyone says, I have this specific regulatory question I want an answer to. And so it's really, it's a fine line, it's a balance in that conversation to ensure that it doesn't go off the rails quickly. And I have complete confidence, Tina, that you can do that. But it's, that is, I think, going to be the one biggest red flag that's going to keep popping up time and time again in each of these types of interactions.
Well, for me, I think, okay, Susan, how are we going to tackle this? How do we see like stakeholders here, the Reagan-Udall Foundation to help FDA as this buffer, and then we have FDA, and how can we think about getting that input from the community and accelerating those that matter, those mature concepts to FDA? I'm thinking that's going to be the place we get to work together, Susan.
I hope so, because we're ready and excited about it.
We have one last question from the audience. Let's chat.
Oh, please.
So my name is, can you?
Yes, we can hear you. Or we could hear you.
My name is Megan Wankura, and I'm very interested in this conversation because I'm about to graduate my PhD, and I'm really interested in having general better dissemination of research amongst different populations. I haven't really thought about the FDA or any of government regulations prior to today, and this has been really informative. And I think a lot of the issues that you're talking about are the same issues I've been thinking about, and it's really interesting, specifically thinking about these regulatory agencies as the ones who have the tool sets to manage the different types of collaborations. And I was wondering if you could speak on literally just how you manage the data and communication of the collaborations you do form. Is this all happening kind of over email, or you talk about the cloud, how infrastructurally is this happening? And I think that social media is so important for dissemination, and it's really interesting to think about how these how things like TikTok and Reddit and LinkedIn, Twitter all overlap with government.
I think that what I can say is the agency is still working to evolve its infrastructure to be able to strengthen its collaborations outside. Unfortunately, we still struggle with ways to engage with even our most critical frontline stakeholders. If I'm trying to do a project with Susan, unfortunately, for the time being, we probably will interact over email until we can build a more robust infrastructure for engagement. And I think it's a critical need for the agency and for regulatory science. That being said, doing our day jobs in terms of being regulators itself. So we definitely need support and help on that front. But I will encourage any of you who are interested in this dialogue or engaging to connect with me on LinkedIn. I'm extremely active in the community. I'll be working with all of our stakeholders today and moving towards a regulatory science collaborative community where we can connect all the collaboration models that are already in place. I'm not trying to create new infrastructure. We don't have the resources to do that at FDA, but I do want to capitalize on the resources and the infrastructure that already exists. And doing our part to do that. So with that, I would like to pause and see if anyone else has any thing to offer on that with that point, or any other points before we close for the morning.
Well, I'm certainly going to encourage people to connect with me and with the SEED office on LinkedIn or through any other platform. So always happy to increase the scope and span of my network and those people that I can reach out to, as well as who might reach out to me.
Yeah, absolutely. Dr. Zhang or Klaus, do you have anything else? Oh, sorry, Leonardo.
I just want to echo that. We certainly do not be afraid to contact, to ask questions. We always provide feedback. And this was absolutely great. Look forward to continue work on this.
Discussion. Looking forward to the next steps.
Yeah, I think that's probably leads us, Tina. We need to wrap up. Yeah.
Yeah.
Again,
Oh,
this is just the beginning of a journey. I believe, we've been talking about this, and also I want to encourage the audience here that, you're still staying here, meaning that you're interested in this. So keep in touch. We would also like to include all of you in the loop. So you can look up my contact and also Tina's contact. I'm the SFB president, easy to find me. So Tina, you want to wrap up?
Yeah, I just want to say in closing, thank you so much to our panelists and presenters today. To those in the room and on Zoom, you can't imagine the amount of effort we spent together as a community to build this roundtable for you all today. We were really focused on the mission of the annual meeting for SFB, the perilous fight to translate innovative research to commercial viability. That's not just for the traditional medical product. Commercial viability, in my mind, also thinks about the ecosystem. And as Klaus put it so nicely, success is a function of time and its people. And so if you want to be a part of this, please connect with us. We're your people to move this regulatory science community forward. Thank you so much for your participation today.
We will keep in touch, and all the panel members as well as the virtual and real in-person attendees. And this is again just as I mentioned in the beginning, this is just the first step, and we'll have a lot of follow-up. So keep in touch. Again, I want to thank all of you, the panel members and all the attendees. Thank you, Tina, as well. Great job.
Thanks, everyone. Be well.
Have a great reminder of the conference. Bye, folks.
All right. Thank you. Yeah.